second mission of the center is to develop new crop of engineering leaders who also conduct research at the interface of biomolecular and physical sciences. So, despite the fact that conventional reductionist biology has given us so many insights into how genomic systems operate, to a large extent, the analogy that comes to mind is magic, Harry Potter. That's why I'm showing the picture of Harry Potter. There's a magic wand, a certain pathway gets activated, and we see what happens, a change in phenotype, for example. But clearly, biological systems, just like any other in this universe, have to obey the laws of physics. And we would like to understand them from the first principles of physics. One of the central missions of the field of physical genomics, or PhysGen, is to understand the relationship between the structure and function of the genome and their role in generating global patterns of gene expression from the first principles of physics. Understand this as the physical system and learn how to regulate the system, again, by applying principles of engineering at the physical level. The investigators at the CPGE, at the Center for Physical Genomics at Northwestern, develop new technologies that span imaging and computational genomics and genome mapping, and deploy those technologies to address several fundamental biomolecular questions, such as how does the genome organized? What are the physical drivers of genome organization? How this chromatin structure of the genome affects transcriptional processes, and how this complex transcriptional processes lead to a specific phenotype and cell behavior. Then the combination of these technologies and fundamental questions are deployed to develop translational applications in cancer diagnostics, therapeutics, regenerative medicine, stem cell technologies, and other applications. Clearly, it does take a village to even begin to address this kind of questions. CPG is fairly small today, but it's growing. It includes 12 faculty from 3 schools and 11 departments, again, spanning several fields of engineering and physical sciences, medicine, and biology. There's also a growing network of external collaborators from Brown, University of Chicago, University of Michigan, and several other universities. The center is benefiting from several high-profile funding programs. One of the recently awarded center grants is from NCI, Cellular Cancer Research, Imaging Research. So now the center operates an NIH-funded center for chromatin nanoimaging in cancer, which brings in 7 very talented investigators from Northwestern, again, who span fields of computational genomics, imaging, molecular biology, and oncology. The center bridges those fields, including nanoscale imaging, computational imaging, molecular modeling, computational genomics, cancer, stem cell biology, and oncology to address some of the fundamental questions in the emergence and persistence and resistance of cancer stem cell phenotype and their role in carcinogenesis. CPG also is part of another network called METNet, Metastasis Research Network, also NCI funded through U54 program in collaboration with MIT, UPenn, MD Anderson, and Dana-Farber. Equally importantly, I should say, is the educational goal of the center. And the center recently got funded by NIGMS to develop T32 Training, pre-doctoral training program in physical genomics, which makes it actually the first, one of the first training programs in the country. The training program includes 38 faculty from 3 schools and 17 departments, and we're welcome 6 inaugural trainees who are here with us in this virtual audience. Last but not the least, I would like to announce 2022-2023 Alan Taflov Fellowship in Physical Genomics. And this fellowship, sponsored by the center, is in memory of a dear friend of several of us here in the audience, Dr. Alan Taflov. Alan Taflov was a phenomenal, extremely talented, and highly influential and recognized faculty at the electrical engineering department here at Northwestern. He had a remarkable career. He was the founder of the computational tools in electrodynamics. So whenever we talk about solving Maxwell equations using a computer, which is done in so many circumstances today, all of this work goes back to Alan's inaugural paper several decades ago. Back then, when he was a junior faculty, he did have a hard time because a lot of luminaries back then were ridiculing Alan, saying that computational electrodynamics would never take off. Computers would never be powerful. It's a useless field. And he was frankly concerned about getting tenure, whether he would get tenure. And of course, he proved everybody wrong. But despite all his successes, he remained, most importantly, a wonderful person. The door of his office was always open. Whenever you passed by his office, he was always meeting with a student, not just graduate students, but also undergraduate students. He was heavily involved in research. He was passionate about training undergraduate and graduate students. He was always willing to help everybody. He indeed had a wonderful life, and we would like to celebrate this wonderful life by starting a fellowship, which will provide 2 quarters of graduate student tuition and stipend. Those of you in the audience who would like to apply, if you are a student, please visit the webpage provided here, and it's also listed at the CPG CPG Center web page as well. So please feel free to apply, and we're hoping to have a wonderful crop of new trainees who will be trained in memory of Alan Taflov. Thank you so much. And with that, I would like to, without much further ado, start the main part of the program. We have 5 fantastic speakers today who epitomize the best about convergence research. The schedule is going to work the following way. We have 3 talks first in the morning, followed by a short break, and which will be followed by a panel discussion. So all 5 speakers will be part of a panel, and every one of us in the audience will have a chance to ask a question. You're welcome to ask a question directly or put it in the chat, either to everybody to see or directly to me. I will be the moderator. I will pick up your questions and I will ask them. If you have any question, and I hope you will, during any of the talks, again, please put them in the chat. You're welcome to share them with everybody, or you're welcome to share them just with me. I will select some of those questions and ask them either specifically to the speaker you want to ask the question to, or everybody on the panel. So we will not do Q&A during the talks, but the Q&A is going to be wrapped into this into the panel discussion as well. And what is a better way to start than with a talk by Alistair Boettiger? Alistair's work really epitomizes what I would say physical genomics is, or convergent research more generally is. Convergence research at the interface of biology, engineering, imaging, computational sciences, and physics. Alistair had a remarkable training pedigree. During his postdoctoral work, he did some revolutionary work at Harvard at the interface of genomics and imaging. Research in was his PhD research. He worked in Berkeley, where he published some seminal papers on cis regulatory sequences that modulate the robustness of gene expression. And in his research at Stanford, Alistair is asking some of the most fundamental and fascinating questions one can think of. For example, how can we understand how cells with the same genome can develop such dramatically different behaviors? Some of the very fundamental questions, and again, addressed using convergent science approach. So I would, last but not the least, Alistair has received a number of prestigious awards, including Bachman, the Investigator Award, Packard Award, New Innovator Award from NIH. I'm sure many more accolades and awards are in the pipeline. So with that, I would like to welcome Alistair Boettiger. His talk is entitled Genome Origami, Differentiating Cells by Bending the Blueprint. Welcome.
Thank you, Vadim, for that very lovely introduction. And I'd like to start with a short apology. I actually have a teaching commitment during the later question and answer that I didn't get to move. So I have planned this to keep some time at the end for some questions. So if you have questions, do please put them into the chat or raise your hands. It's not an incredibly large group, and let us I look forward to the feedback. So as Vadim introduced, I got into this question of the physical genome from a love of understanding the cis regulation, or the control of the genome, how a single genome can produce so many different cell types. And we know this actually happens not by having an incredibly diverse number of genes. Genes make up still a relatively small part of the total sequence of complex genomes like our own, but by reusing those genes like diverse Lego bricks in different combinations throughout development to make up different cell patterns. So an individual gene might be used in combination with some other factors to make a limb, and in different factors, for example, to make the brain. And the information to specify these diverse expression patterns and the diverse expression levels that go with them is written in the rest of the genome, the non-coding portions, which we call enhancers. And then the fascinating property about enhancers is their distributed nature. So while they only regulate genes on the same chromosome, which is what we mean by in cis, they can do so at remarkable distances. Many lie thousands to even millions of base pairs away. And a fascinating question is then to understand how this happens across such great distances and how it happens with specificity. And early geneticists stumbling across this problem had postulated a role for the physical folding of the genome in explaining these genetic interactions. But it hasn't been until more recent times in the last decade that we've really been able to address this question systematically as a field and map the remarkable physical structures of the genome. So one of these fascinating questions, why genes can regulate at substantially long distances, and how, despite being able to do this, they have specificity at these long ranges, has been partially answered by the discovery of these features we have called tags. So if you map genome-wide, the frequency at which any piece of the genome interacts with any other piece, so this will be very familiar to some of you, you find a box-like pattern where there are high frequency of interactions within the domain, and then separated by sharp transitions where elements inside this portion of the domain interact less frequently across the border. And we'll look at a couple more of these style of maps. So with the orient, this is genome position along the X and Y axis. So naturally the diagonal is bold because everything interacts with itself, and then the frequency at which it interacts with other points along the genome is indicated by the color. Nonetheless, there are some limitations to mapping genome structure only in this type of pairwise contact frequency approach, which we've been able to do with next generation sequencing methods. It is a map of pairwise interactions, and it is a map of only average pairwise interactions. So this doesn't give us a clear picture of what one of these topologically associated domains actually looks like inside a cell. What does this physically represent? It's actually a statistical pattern as far as we can tell, this population average. We can't detect three-way interactions in this approach. We are only mapping the frequency of two things, and that has to do with the chemistry of how it's done. The sequences are actually ligated together. It requires a large number of cells to create this sort of map. There are indeed single cell variants that some of you will be familiar with, but they're inherently quite sparse. So you don't map out structure, you map out a subset of interactions. By requiring large cell numbers, we lose access to the cell-cell variation, which is one of the things we're especially interested in tracking changes in structure across development, is how does this start to differentiate. It's also quite difficult to use in tissues and embryos. Just many of our favorite tissue types, it is challenging even to acquire the amount of material needed to perform a sufficiently high resolution high-C study. Finally, the approach discards much of the rest of the cell. So we've lost the spatial information in the tissue by resorting to a bulk sequencing approach, as well as other cofactors you might be interested in looking at, most notably, for example, the RNA expression. And lastly, it's quite expensive, despite the fact this is enabled by deep sequencing, and deep sequencing is generally a very cost-efficient way to generate data. This is one of the more greedy approaches because your coverage scales with the square of your genome size, not the linear genome size. It's common to sequence into the billions of reads, and that starts to add up, especially for many cell types and many mutants. So we've been taking a different approach to study the nuclear architecture around our favorite genes, which is conceptually quite simple. We start at the same point, use these high-C type approaches. We first chemically fix the nucleus. We freeze the cell in whatever structure it was last in, so that we can have some time to study it. And we then hybridize probes to the genome. These are little DNA oligos, and each of these DNA oligos carries a fluorophore. And this builds on techniques that have been a driver in the field for a long time, called fluorescent in situ hybridization. And here we've exploited the advances of modern sequencing and computation that we use instead synthetic oligos, so we can ensure that the binding sites are unique and the binding kinetics, the melting temperatures, etc., are very uniform across the domain. Nonetheless, if you tile your region of interest with probes, as I've illustrated here, and then we put it under the microscope to see how it folds, it will reveal not this beautifully folded polymer like I've sketched here, but see instead one of these little diffraction-limited blobs. And the problem here is not that our magnification is not high enough, but that in fact we've reached the physical limits of light microscopy. We are trying to see the twists and turns of this 10 nanometer chromatin fiber, and we're using some 500 nanometer light, and the diffraction of that light has just blurred it all together. We can circumvent this with a variety of approaches these days, available in molecular biology, and one that I embarked on my postdoc to learn, the stochastic optical reconstruction microscopy, can indeed reveal substantially more structure than these diffraction-limited images. Somewhat disappointingly, though, for the study of these cis-regulatory interactions, we have not penetrated with STORM the resolution to resolve the actual twists and turns of a single fiber. In some places, maybe you can see it as a fiber, but in others, it's just too dense. So to get around this problem, we have an alternative approach, which is to replace each of these floors are actually small clusters of the floors with unique DNA sequence tags, and then address the tags sequentially by adding complementary oligos tagged to the fluorophore. This also creates a large spot on our detector, but we can know the sequence of interest lies still at the symmetric center of those points. And we can fit this quite nicely computationally. So here's what some real data actually looks like. You get these large diffraction limited spots on your detector. We can fit these to sub-pixel accuracy in 3 dimensions. We record that position, and then we use a strand displacement reaction to strip off the probes. This is important as the sample now goes blank, and we can add the next set of probes. We get another spot in almost the same exact place. If they were both on at the same time, you wouldn't see, you still have a unimolar distribution, but because there's only one at a time, we can still fit the centroid here with high accuracy. This sequence then repeats in the example here for a further 65 rounds until we've traced out the full trajectory of the polymer. And because it's somewhat difficult to persuade a graduate student to sit there and hybridize on and off 65 rounds, we've roboticized this process, so it happens in an automated way on the microscope. And in many ways, what's happening on the microscope is actually quite similar to what's happening inside one of your sequencing machines. We have rounds of adding chemicals, fluorescent probes, imaging them, removing them, and proceeding to the next round. This super resolution reconstructed image now has much more structural detail than our diffraction limited spot. It also has a lot more genomic detail. And rather than look at this only as a polymer, we can also look at this as one of these 2D maps, which helps with the 3D projection issue here. Well, I'm plotting positions of the barcodes on each side, and then the distance between them is indicated by the color. And position or barcode number, I'll remind you, reflects position along the chromosome. So we call this approach optical reconstruction of chromatin architecture, or ORCA for short. It's a homage to storm, which is also optical reconstruction, and a fish being ORCA being another marine organism, just a little bit larger. And I'd like to make a shout out to a number of colleagues in the field who have also been exploring these powers of sequential hybridization to really study genome structure. It's a quite robust and effective method, and it scales very nicely in throughput, much more so than we were able to do previously with Storm. So we're likely to see much more in upcoming years. I showed you conceptually how it works, but we might yet wonder, do we measure the same properties that have been mapped with the molecular and sequencing-based approaches? So if you take many of these individual polymers mapped in a single experiment, we can plot the frequency any 2 balls are nearby one another. And that's what we're doing here. As a map, this is the frequency any 2 are within 150 nanometers. We're intentionally trying to mimic here the structure of one of these high-C contact assays that measures contact frequency and normalized reads. And I was blown away by the quantitative agreement of the 2 approaches. After all, both have done numerous unspeakable things to the cell in between when it was a living cell and when we finally got the data. Here we've denatured the genome, hybridized these probes too, and walked through one step at a time. In this approach, you've also cut the genome into many pieces and ligated it back together, then sequenced the whole mess and normalized it with some matrix balancing magic. Despite these dramatic differences in approach, as well as how they affect the sample, we see very similar structures here. The boxes that we call tags, or the boxes in boxes, the loops and stripes. What do we learn from this approach? In addition, that we haven't learned from being able to provide the whole genome structure on a population level. We alluded to some of the limitations to start with. Now that we have single cell structures, we can ask just how variable are these? And are we looking here at a stable structure like a protein organization or something different? And as I think you can appreciate from these images, there is remarkable variability in the folding of individual domain cell to cell. Here we're just showing 2 examples and their corresponding distance maps. And it is only in ensemble, when we start averaging together several hundred to 1,000 of these, that you trace out a frequency map across the population that starts to converge to the population statistic measured by Hi-C. So this is not a structural polymer that's folded into some stable number of 6 or 7 domains. It is in fact a highly dynamic polymer that is changing significantly between cells, presumably over time. And what we see here are the frequencies at which those different positions and different boundaries are actually adopted. So unlike chromosome territories, unlike many of our stable protein foldings, the large heteropolymer that is our genome does not adopt the same structure. While this has been very instructive in understanding how TADs link to gene regulation and what the physical basis of this contact frequency actually is, there are several peculiar exceptions that we haven't been able to explain with the population level statistics. And this is what I would like to spend the rest of the time on today in talking through some of the examples that come up in development where we genetically link distal regulatory elements that hop over these boundaries that we just examined that previously have been in other cases correlate to separating regulation. And this is the work of a talented grad student in the lab, Su-Chao Hung, and he's been studying this locus, which was first characterized by Stefan Mandelos' lab and followed up in collaborations with his postdoc and now Professor Guillaume Andre. In this domain, a distal enhancer several hundred kilobases away is important for driving the expression of this gene PIDX1 in hind limbs. And if you don't properly activate PIDX1 in the hind limbs, you don't properly differentiate the hind limbs from the forelimbs, and you have these partial transformations of legs and ankles that look more like arms and wrists. So it's a feature that was originally actually identified in human genetics, and this enhancer is conserved across many of the down to chickens. A surprising observation about this enhancer is that the transcription factors that bind the enhancer to activate this more distal gene are not limb specific. So if you just probe the activity of this enhancer in an isolated assay where you fuse it immediately upstream of a LAC-C transgene, the enhancer drives expression in both hind limb and forelimb. And it is only in its native genomic context, where it is deeply separated from the PIDX1 gene, that you get this specific activation in hind limb, which requires a hind limb specific refolding of the genome to increase these interactions. So Su-Chao was asking, how does this happen? How do you bypass, how do you refold in a different structure in purely the hind limb? And how do we do this in such a way that we can bypass these existing TAD boundaries and CTCF sites that have been previously shown to block distal enhancer promoter communication? And Su-Chao started with 3 hypothetical models for how physically you might fold a genome to enable one of these bypass interactions. His first hypothesis was that the region might loop out from the domain, that while these domains may be relatively stable, the regulatory sequences being quite flexible may leave the domains and interact. The second hypothesis, as we based off of the sort of single cell structures we just showed you, is that the domains are quite dynamic and that they merge some of the time, and that these merging events would be an opportunity for the enhancer and promoters to interact. And this was my preferred model going into our analyses. Third, it was possible, imaginable, that while the domains remain relatively separated, that regulatory elements could all meet in the middle if they all sort of stack around a central hub. So this idea of stacking the domain borders was his third hypothesis. While these look different in their physical structure in single cells, because as we've already seen in our dynamic structure of the genome, the enhancer promoter interactions are transient events. There's a diverse ensemble of structures. They would be lost in the population of cells imaged at any individual time from all of the other conformations. And each of these cases would look the same by Hi-C. What we need to do instead is to pull out only those that are facilitating the interaction and ask what physical structures are associated with these to distinguish between the three different mechanisms. So this is what Cigall proceeded to do next. To use this approach we just described of the optical reconstruction of chromatin architecture and image the structure in both hind limb and forelimb cells. We're showing just an example here. We've highlighted the gene and some of its known enhancers. And this one here, PEN, is one of interest that is itself not tissue specific, but makes that tissue specific bypass. If you average these together in several tens of thousands of cells from both hind limb and forelimb, you can see that the contact frequencies on the whole look qualitatively similar in the individual domain structure. But if we subtract these two, we can see, well, the qualitative features and borders remain stable. The quantitative strength of these borders changes between the hind limb and the forelimb. And this is something which is another advantage of microscopy, because these data are not in arbitrary units of normalized read counts specific to their individual experiments, but a fraction of cells under a particular nanometer of physical distance threshold. It is much more interpretable to look at these subtractions. What you see here then in blue are regions where there's actually more contact in forelimb, and overall the domain is actually more interacting in forelimb, except at a few specific points. So I've superimposed the TAD boundaries here. And if we look at this interaction, for example, which connects the PIDX1 gene to this distal PEN enhancer, we can see that is indeed more frequent in the hind limb. But we have an even deeper quandary to explain, because not only in hind limb is it passing over these other CTCF sites and TAD boundaries, but those borders that it hops over are actually stronger in the hind limb cells. It's something we'll come back to in a moment. Now on to testing our three hypothetical models. First model is the looping out of the domain. Well, Cigall can find some examples of structures consistent with this type of behavior. We have a relatively intact PIDX1 domain here in red, a PEN domain in blue, and kind of sticking out the back of these two domains. You can see right here the interaction between the PIDX and PEN enhancer. This is still a relatively small fraction of the total enhancer promoter contact events that happen in this type of structure. So it does not explain the majority of what is going on. It's also happening at a similar frequency in hind limb and forelimb, so it's really not explaining the hind limb specific regulation. My second model was to look at the cases where these domains merge. We know there are boundary factors that are important in establishing the separation between the two domains. I didn't walk you through that, and that these things probably bind stochastically, and we've seen in our previous imaging a fraction of the cells merge the domains. We can find many examples of this. So here the red, blue, and purple domains are all relatively intermixed with one another. We asked, does this help increase the probability of enhancer promoter contact? So we take all of the maps that lack installation between PIDX and PEN. There's this type of merger. And we look at the fraction of events that have enhancer promoter contacts. And we do find some, but again, it's a minority of the total enhancer promoter contacts that are happening in the data set. We ask, so does this merging actually improve the probability at all of enhancer promoter contacts? Maybe the merging is rare, but the fraction of bump associated with this merge is enough to increase the expression. If we actually compute the relative risk, the effect of the correlation between being merged and having a contact, we find actually really no correlation at all in the hind limb. If anything, the merged cells are ever so slightly less likely to have a contact. Finally, is this domain merging more common in hind limb than in forelimb? There's pretty similar frequencies in both. If anything, it's a little more common in forelimb. So this is not appearing a good model to explain the cell type specific interactions of this distal enhancer and its promoter. Which leaves us with our third model. There are cases where all of the enhancer elements, including PIDX and PEN, have met near the middle of the domains. We asked, how frequently does this occur? And we looked among all cells in which there is an enhancer promoter contact event to start with. There are two boundaries in the domain. If we condition on the cases that interact with either one of those boundaries, it's actually a surprising number of the total interactions. So if we ask, does this increase the probability of enhancer promoter contact in the relative risk score? We see it does. It's quite significant, though moderate effect. And is it more common in hind limbs? Yes, indeed, it is more common in hind limbs. So, in summary of this part, the way enhancers in development, at least in the case of PIDX, have managed to bypass boundary elements in between is not by ignoring the boundary element and merging in a fraction of cells. That does happen. Boundary elements are not impermeable, but this is not the mechanism that specifically increases the interaction in hind limb. Instead, it is by embracing the boundary that the enhancer is able to hop over this border. And in the case of PIDX, it actually is able to embrace two boundaries along the way, leaving preferential separation between the domains. What mechanism actually drives this? Close with this part, i.e., what properties can we give a simple minimal model? As physicists, we like to be able to have minimal model reconstructions of things to see that we actually understand the basic properties involved. And here we built off some existing models in the system, which treat the DNA polymer or chromatin as a polymer fiber. And we impose on it, in addition to Brownian motion, one more property, that there are special proteins that can bind this polymer, like cohesin, and extrude a loop on this polymer, but they will rest at particular sites, like these boundary elements, which are represented by CTCF. And we started with this model as it was previously shown to be a good model of creating these pads in the first place and explaining the preferential isolation across the domains. And we asked, does this model also have room to explain this bypass of these borders as well as the existence of these borders? So if we watch this in a dynamic version, you can see here these cohesive molecules are binding. Where they extrude a loop, I've colored the loop. And otherwise it dances around. You can change the couple properties in this model, like the processivity or loading or unloading of cohesin. We find changing none of these makes it sufficient to explain the difference between hind limb and forelimb. But if we play with the binding probability of these blocking sites of CTCF, we can indeed explain this difference between hind limb and forelimb. And here's starting with just two examples. Actually, when this border factor is at high concentration, the probability that it then captures one of these cohesins is also higher, and rather than that the cohesin walks right by the site. And consequently, these CTCF sites get pulled to the middle by these cohesin loops, and they collide more frequently all in these cohesin chained bridges, where this happens at less frequency in the case with only with lower concentration. And we proposed it as these actual events of multi-loop bridges that are allowing the elements, the enhancer, to specifically access the promoter. If we show not just individual examples, but the subtraction frequency, we can see, much like we saw in the data, this property where the enhancer-promoter interaction is increased when the boundary factor is at higher concentration, even though there are several borders that it had to hop over here, where the borders are actually stronger at the same time. And there's a little bit of a 3D picture of how this happens and what it means for our understanding of border elements. We've seen that while border elements do indeed prevent interactions across neighboring TADs, for elements that are situated at the borders of adjacent TADs, they can facilitate these cross-TAD interactions. And I think the structural picture is very valuable in visualizing this a little bit better, that multiple border elements, as indicated by these red triangles, lead in extruding multiple loops together, lead to these structures where the border elements are all pulled up on this axial structure of the chromatin, enabling the distal enhancer that has to cross several borders to regulate or be in close proximity where it can regulate the enhancer on the far side of the border. Nonetheless, these border elements still separate genes that are out on the loops that are farther from the border. So while this gene is nearby in linear space, as is the case for one gene here at this PNX1 domain, it is on average further away in 3D space. And the stronger this border, the more likely it will be caught in the middle, as well as the facilitating this interaction while preventing that one. And the same is true for distal enhancers that might need to be separated from these promoters. In the interest of time, I think I will close out with that. This is a final example of testing these interactions by removing CTCF in a different cell system. With that, I'd like to thank Alistair Boettiger, who has led this work, our sources of funding, and all of you for listening. Thank you very much.
Thank you, Alistair, so much. So, and this first talk is a little of an exception because Alistair has to leave due to his teaching duties, which we all understand, of course. So, if you have any urgent questions, I think right now might be a good time to ask them. For everybody else, we'll delay questions for the panel.
I'm happy to take follow-up questions by email. I know I took a little longer in running through it than I.
Well, so I had one quick question. So you talked about three different possibilities, I guess, for the boundary, domain boundaries, right? For the formation of domains. What do you think drives this different possibilities? Is it primarily regulated enzymatically, or there are some, I guess, polymer physics-based differences that may evolutionary result in some of those configurations?
Yeah, I think fundamentally the diversity of structures we see is reflecting the physical mechanisms that are folding the genome. And I think some of our intuition as biophysicists has been skewed a little from our interpretation of polymers, which lie in a unique state. Many of our protein polymers with structure lie in an interesting state where there actually is one well-defined global minimum. But that this is not generally true of heteropolymers and not generally true of the chromatin, so that there are multiple low energy states for the for the structure of this protein. And that's further reinforced by the fact it is not only folding based on affinities, which does seem to be happening, but also based on the action of these motor proteins. Understanding that heterogeneity then makes an additional challenge for understanding gene regulation. We haven't just a single structure that you can be, I now understand the folding of these two domains brings them both near the active site, and that's why they both influence. The structure is more dynamic, but that's also something that the development can then play with and tune the bias, which is, I think, what we see happening in this Heinlein forelimb example, to spend more time in the state where this enhancer can access the promoter, or less time in the state, which is not enough, apparently, to kick it off into a proper expression.
Thank you. And if you have just maybe like a couple of more minutes, there's a question from Amy Strom, who congratulates you on your great talk. And her question is, what length of sequence can you cover with ORCA? And I guess a follow-up question is, does it work with repetitive sequences, such as what you find in heterochromatin or telomeres?
Yeah, I mean, these are great questions. Because the probes are synthetic, we can cover arbitrarily sized domains. Actually, some of the first work by Stephen Wang's group started at the chromosome scale. And the challenge has been primarily to drive the resolution down for smaller elements to keep good resolution. They're much more sensitive to the vibrational noise and the fitting accuracy of the individual spots. And there, the lower limit is, for us, it's been around 2 kilobases. The upper limit is the whole chromosomes, but the resolution you get the chromosome depends on your patients. A nice caveat is seen in some beautiful work recently from the Young Lab and Kai Labs. You can exploit the fact they overlap less frequently on the whole chromosome scale, and you can indeed use combinatorial barcoding on this chromosome scale to get at megabase resolution whole chromosomes, which is also of interest. Repetitive sequences are a challenge.
Right. Well, thank you. And there's one more question from Adam Williams. Adam, would you like maybe to perhaps voice your question, ask your question?
I can read the question too. Adam asks, this is a larger organization to facilitate a single promoter-enhancer interaction. And are other enhancer-promoter interactions maybe piggybacking off this event to change their expression? I think this is a great question. It's not one I think we've nailed down entirely. It is possible to purely speculate that by increasing the boundary strength to the adjacent TAD, PIDX is achieving two things at once. In these cells where it wants to turn on, it's isolating itself better from enhancers that might drive it in the wrong cell types, as well as facilitating contact with this one quite distal enhancer. But clearly further work is needed to really understand all of the evolutionary logic too in making these changes.
Well, thank you so much. I profoundly enjoyed your talk, and good luck with your teaching. If you have a chance to get back, happy to have you.
Will. Yes.
All right. We're going to give you virtual applause. Thank you so much. Thank you. All right, and now it's time to go with our second speaker, Ursula Jakob. We'll give the second talk. So Ursula is a professor at the University of Michigan, and she has also has been looking and studying the genome processes from a variety of perspectives. Perhaps one of the most significant thrust in Ursula's lab is the studies of the role of oxidative stress on host defense and aging, something that I think all of us can relate to. These are the processes that are so critical in response to an infection, for example, bacterial infection, but more generally, these are some of the most critical aspects of aging. And we're hoping, Ursula, that you will find a way to defeat aging so we can live forever if we wish to do so. Ursula has had a remarkable career and a luminary in the field, which has been recognized by multiple awards, including being elected a member in the German National Academy of Sciences and a fellow of American Association for the Advancement in Science. And without further ado, I would give the virtual microphone to Ursula. Welcome.
Thank you very much. I'm going to share my screen. Just making sure you can see it. Which, do you see the presenter view or the other view?
We see the good one, the one that we, not right now. Right now, we see the
okay, here we go.
Super. Second to do it was that's the one.
Okay, perfect. So I'm taking you a little bit more into biology now from genome structure, but I want to raise your awareness about how small changes in the environment, small changes within even an isogenic pool of cells can have, or organisms can have, very long-lasting longitudinal effects. And that's something that we are very, very interested, how small, often you call it stochasticity or randomness in processes. And we are trying to figure out how do they develop, where do these the randomness come in, and what are the long-term consequences of that. And the model that we picked is aging and age-associated diseases, and I tell you in a moment why. So you probably all thought about at some point or the other how long we're going to live, and what are the factors that play a role. And it's clear that there's a genetic component. There's in the family of Helen Reichert, they're all 4 siblings became centenarians. They had 3 mutations that researchers associated with this long-lived phenotype. So that's the one, that's genetics. The second part is, of course, environment. I mean, if you have a gun-wielding younger brother and you don't survive that well, then that's environment, that's what the environment does to you. So, but then there's a third component that affects lifespan and longevity. And this is best studied in an isogenic organism. And so one of the ones that we are studying is the small roundworm C. elegans, which you probably know is a hermaphrodite, and it produces about 200 offspring, and they are genetically identical. And we can grow them under exactly the same conditions on a plate, and then monitor how fast they die, when do they stop laying eggs, and so on. And so what you would think, if lifespan, aging, age-associated diseases was all just determined by genetics and by the environment, they should all drop dead at the same day. So when you look at their lifespan, however, you see that some of them die within 2 days and others live for 25 days. And so this is what researchers in our field call the stochasticity of lifespan, and where we just don't know exactly where it comes from. Because again, they are genetically identical and maintained under exactly the same conditions. And this is what we are very interested in. And we're not only interested in this in regards to C. elegans, for instance, but also clonal populations of stem cells, for instance, where you also know that within a clonal population of stem cells, there's a huge variability, and some differentiate and the others don't. So what are the factors? So my interest has been for a long time in reactive oxygen species. And the reason why I think they play such an important role is because they're constantly produced in every aerobically living organism. And what's very important is, of course, reactive oxygen species are toxic, they should be avoided and so on, but we need them as well. We need them for metabolism, growth, differentiation. Now, we also know that metabolism and hormonal cues trigger reactive oxygen species production, particularly H2O2. H2O2 levels are also affected by by the bacteria that are being the microbiome, as well as exogenous sources. Now, the downstream effectors of reactive oxygen species are redox-sensitive proteins. And we now know that basically every pathway in the cell has at least 1 or 2 redox-sensitive proteins that adjust the pathway to the redox environment of the cell. And those proteins affect metabolism, growth, and differentiation. So now you can start thinking about if there are small metabolic changes within an organism, maybe a little bit more colonization of bacteria, you get a little bit more H2O2 that affects then these redox sensitive proteins, which then affect these downstream processes. And in a feedback loop, you make potentially even more of these reactive oxygen species. So this was one of the ideas how a small change within an organism can have ultimately long-term and profound consequences. But how do we test this? Now, the real breakthrough in our field came with the development of redox sensors. These are ratiometric chromosomally encoded proteins that we now have in embryonic stem cells, in the C. elegans, we have mice. So we have all these different systems. And what's so cool about them is they're ratiometric. That means it doesn't matter how much of the protein we express or whether the expression changes over time. As long as we have some protein, we can look at those 2 excitation maxima, and they relate to either the endogenous H2O2 levels or the levels of the redox coupled GSHG over GSH, so the glutathiones. And so with that, we can then do live cell imaging. We can sort the cells, the organisms, and then test how does intercell or inter-individual differences in the ROS levels affect downstream processes. So that's a very, very cool technique. I show you most of our data with the GSH, TSHT sensor, but we've reproduced many of them also with the H2O2 sensor, which is as expected. Since you increase levels of H2O2, you also change the ratio of the redox couple. So we did this in C. elegans. We had a synchronized population. Every one of these little symbols is a worm, and we just monitored the redox state over time. And we found that during the adulthood, as they grew older, the worms became more oxidized. That's something we knew. I mean, that's a 50, 60, 70-year-old hypothesis. What was very unexpected, however, was the very early stage in this organismal development, in larval development, where we saw also much higher levels of reactive oxygen species. And this became even more important, this large inter-individual variation, with some being very oxidized and others being very reduced. So this is very early in development. And so we thought, maybe at this stage, these ROS levels can start putting events and processes into motion that ultimately then contribute to the big lifespan variation that we observe. So we sorted those worms. We did a live sorting and then analyzed various longitudinal outputs. We looked at redox homeostasis, we looked at stress resistance, and we looked at lifespan. And under all of those conditions, we always made the same observation, that the worms that experienced endogenously slightly higher concentrations of H2O2 and were more oxidized during this larval stage were better off. So they had a more balanced redox homeostasis. In fact, they switched and became more reduced as they aged. They were more stress resistant against a variety of different stresses, significantly when you compare the red versus the blue curve. And they were also longer lived. So this was really cool because it suggested to us that you can have some early life events that affect very late events, events very late in life. So then we thought, okay, before we look at the mechanism, let's try and switch this. Can we actually switch the ones that were longer lived to shorter lived if we just change the redox status during that particular developmental period? And that's what we can do. We can take the oxidized worms in red and treat them with either more oxidant, as paraquat, or with a reductant, which is N-acetylcysteine. And so when you look at the red curves, we see that nothing changes when we add more oxidant, but when we add more reductant, they become actually shorter lived. Conversely, when we took the reduced worms, which were originally shorter lived, and treated them just for 10 hours with an oxidant, they became longer lived, while the reductant didn't really have a much effect. And then we decided, well, can we make the whole population longer lived just by giving them a boost of oxidative stress during early development? And this is what is shown here, where we took the entire population, treated them with paraquat only for 10 hours during their larval stage. This increased their redox status, as you see in the inset, and it also increased and extended their lifespan. So this suggests that there are events that happen early in life that are set. And even if you change the redox status, and I want to go back to that and point this out in the upper panel at L4, when they're just becoming reproductively active, they're all very reduced, the worms, and there's no inter-individual difference. So something that happens must be then resistant to any other changes in the redox environment. So these were all our conclusions that we had. So the question then was, what causes this change that has such long-lasting effects? So it turns out, so we took the oxidized and reduced subpopulations and did RNA-seq analysis. And we expected some longevity pathway genes to be upregulated, anything that would fit to our idea. Yes, they are more stress resistant and they're longer lived. But that was not the case. There was only about 200 genes that were differentially regulated between those two subpopulations. But what it became clear was that the expression pattern was very similar to the expression pattern of a histone modifier or of C. elegans that were defective in some in histone modifiers, specifically the histone 3 lysine 4 trimethylation. So we saw this overlap with strains that lacked components of this modifier, and even more overlap, as you see below, with the readers of this histone modification. So that suggested that our oxidized worms seem to look, in terms of gene expression, very similar to worms that have been previously shown to have lower levels of this mark H3K4 trimethylation. And indeed, when we looked at global H3K4 trimethylation levels in the oxidized subpopulation, we saw reproducibly about a 30 to 40% decrease in that level, which suggested to us that the redox sensitive the redox sensitive component is actually a downregulation of H3K4 trimethylation. Of course, we didn't know whether this was just a byproduct or the cause of all the long all the changes that we see in later stages. So what did we know at that stage about H3K4 trimethylation in C. elegans? Well, in C. elegans, this modification, which is reflective of transcriptional memory, is actually set very early in life, which fit very nicely to our data. It's set before the L3 larval stage and doesn't change any longer during the adulthood. So what's set at that stage is set. If it doesn't set, it doesn't get set anymore. So that fit very nicely. It was also known that it causes very few global changes in gene expression, despite the fact that it's reflective of transcriptional memory. And Brunet and co-workers showed that worms like our oxidized subpopulation were longer lived. So this was really suggesting to us maybe this compass complex, which is consisting of the methyltransferase Z2, ASH2, and some co-proteins, is redox sensitive, causes the downregulation of H3K4 trimethylation in those worms. Once that's set, it will not change, and that then will persist into later stages and ultimately cause lifespan extension. So then we thought, well, let's have a look whether downregulation of members of this compass complex have a similar phenocopy our oxidized subpopulation, and they do. I mean, we saw we downregulated H3K4 trimethylation. They were indeed much more heat shock resistant. They were also more oxidative stress resistant, so very similar to what we've seen with our oxidized subpopulation. And of course, and in agreement with what Brunet had already published, they were longer lived when we depleted the methyltransferase Z2 by RNAi. So that all fit together. But then we thought, well, if we now deplete H3K4 trimethylation genetically by downregulating the members of the compass complex and combine that with our sorting, we should no longer see any difference between the oxidized and reduced subpopulation if this oxidative downregulation of this mark was truly responsible for what we've seen. And so this is what we did. We took our redox sensor expressing worms, we downregulated the compass complex, we reduced H3K4 trimethylation levels, we then sorted the worms again, and that was very important. We did not see any effect on the overall redox distribution within these worms. And that was important because that told us that ASH2 and Z2 is actually downstream of the redox, of changes in the redox environment. When we then sorted and analyzed those two populations, which showed these changes in heat shock response and survival and longevity, the moment we did this in the background of an ASH2 Z2 depletion strain, we no longer saw any difference in between the oxidized and reduced subpopulation, suggesting that when they have no longer already have reduced levels of H3K4 trimethylation, that this is ultimately necessary and sufficient to promote these differences that we observe between the oxidized and reduced subpopulation. So based on this, we concluded that a synchronized isogenic population of organisms such as worms at a very early stage in life have already different amounts of reactive oxygen species that are endogenously produced, maybe because they move a little bit more, they eat a little bit more. And that increase, this very transient increase in reactive oxygen species, puts its mark down by affecting the COMPAS complex, and with that maintains these lower levels of H3K4 trimethylation, which are then responsible for increased stress resistance and longer lifespan. So this is in effect a naturally occurring, as we call it, a hormesis effect. So what I didn't show you, but what you might find interesting, is that this is evolutionary conserved. We see the same stress sensitivity for H3K4 trimethylation in HeLa cells. We also see it in embryonic stem cells, which is particularly interesting because H3K4 trimethylation regulates actually the expression of the stemness factors in stem cells. And it's known that oxidative stress is important and involved in triggering differentiation. And this is what we are investigating right now, whether maybe this downregulation of H3K4 trimethylation upon oxidative stress in those stem cells is what's ultimately driving differentiation. We know the target, it's the methyltransferase. It has a redox sensitive zinc center that forms 2 disulfide bonds that transiently inactivate the enzyme. And then once reducing conditions are restored, the methyltransferase works again. So in the second part of my talk, I would like to give you a little bit more idea how changes in this histone modification ultimately affects lifespan and age-associated diseases. And in this part, I mainly focus on age-related diseases. And the reason why we went to age-associated diseases is because of the geoscience hypothesis that strategies that slow the aging process or extend lifespan also typically slow the progression of age-associated diseases. And so we thought that would be really interesting if early events in life already can define whether an organism is more or less predisposed to late onset neurodegenerative diseases such as Alzheimer's or Parkinson's, which we know are related and are affected by similar genetic pathways when we look at that than lifespan. You probably all know that these neurodegenerative diseases are caused by proteins such as amyloid or alpha-synuclein that unfold and misfold and eventually form these beta sheet oligomers that are neurotoxic. And the hallmark of all these diseases is the accumulation of fibrillar aggregates. Again, we've done many of our experiments in C. elegans, but are now moving them also into mice. And so what we have as our proxies, we have C. elegans strains that express these toxic proteotoxic proteins, either A beta 1 through 42, which is accumulating in Alzheimer's disease, or a polyglutamine extension protein that accumulates in Huntington's disease. And we have them either expressed in the muscle cells where the proteotoxicity causes paralysis, or paneuronally where we see then also cell death or paralysis. And so very early on, my postdoc, a very talented guy, Brendan, he did the very same experiment that we did with our lifespan. We treated the worms that express the A beta to paraquat for 10 hours during the L2 larva stage. And what we found was, and that was very significant, we found that the worms that experienced this very short pulse of paraquat, of oxidative stress during larval development, showed a much more, a significant delay in the paralysis, in the onset of paralysis. And that this is also mediated through the COMPAS complex became clear when we did the same experiments, but deleted or depleted one of the members of the COMPAS complex, SET2. In the depletion strain, we already see that strain shows a delay in paralysis. So this is very similar to what we see with the extension lifespan. But now what we also see is that addition of ROS does no longer change any of that effect. So again, this is very consistent with the idea that ROS downregulates SET2, and with that we have downregulation of H3K4 trimethylation, and in this case that causes a resistance to proteotoxicity. This is also the case for some other models of proteotoxicity. This is a chronic model of A beta, where you see that the downregulation of this mark again significantly delays paralysis and toxicity. And this is the Q40 worm, which is the Huntington model in C. elegans. And this was exciting because we then also saw, and this came out only a year or 2 years ago, that targeting the histone K4 trimethylation complex is also has been shown to help treating cognitive defects in mouse models of Alzheimer's disease. And so what these researchers found, both in the mouse models as well as in patient samples, human patient samples, the patients with Braak stages of 5, which is very advanced Alzheimer's, all had higher levels of H3K4 trimethylation in their brain tissue. But when they did, it's similar to the mice here, but when they then treated the mice with an inhibitor of WDR5, which is one of the components of this compass complex, and looked at cognitive tests, they found that this cognitive decline that they see in those Alzheimer's mice can be fully restored by using this inhibitor of the of H3K4 trimethylation complex. So this is something that we would like to go into now more, and also see whether early life events in these mouse models of Alzheimer's disease can have these significant longitudinal effects and might actually delay the onset or lessen the symptoms of Alzheimer's disease. So what is the pathway? So this is just a few more slides to show you what we think the mechanism is and how H3K4 trimethylation has these effects on lifespan and age-associated diseases. We tested some of the longevity pathways that are known and that are conserved, and on heat shock factor 1, which is a key longevity factor that's already known to protect against proteotoxicity. And we did this because we found that, and we still don't know why that is, but we found that the global reduction in H3K4 trimethylation levels leads to a very, very strong and improved response to stress conditions such as heat shock. And so you see this both for our oxidized subpopulations that we know have lower levels of H3K4 trimethylation, as well for our genetically depleted C. elegans strain, that when we stress them with heat shock, which is the trigger for the heat shock factor, we see much improved and much augmented heat shock response. And you can also see this in worms that express one of the target genes, HSP16.2, that upon exposure to heat shock, they make much more of the protein compared to the empty vector control. So these results suggested that HSF1 is either more active or increased in the amount, and that's actually the case. We see much more HSF1 to be made in those cells. And so we thought, well, if this is essential for our phenotypes, if we knock down HSF1, we should no longer have any protective effect by depleting H3K4 trimethylation. And so that's the case. So we see this is in black, the wild type. In the green, when we deplete ASH2, we see the delay in paralysis that we've seen. So they get paralyzed much later. When we deplete HSF1, the heat shock factor, they are much more sensitive, but now ASH2 depletion has no longer any effect. And so we concluded from that H3K4 trimethylation, the downregulation, triggers HSF1 activity or the amount of HSF1, and that now, by whatever mechanism, increases the resistance to proteotoxicity. So this is something, it's a more fundamental question, of course, in the field, how HSF1 can protect against proteotoxicity. And what's also known for HSF1 was that it often worked primarily through development, and it wasn't clear why that is. So for those of you who are not familiar with the heat shock factor 1, it has a lot of genes that it's regulating specifically upon exposure to heat shock. But as you might remember, we didn't find any heat shock regulated genes in our oxidized subpopulation. And neither did people find it when they looked at RNA-seq of ASH2 and Z2 depleted strains. So it didn't look like that HSF1 was constitutively active in producing heat shock proteins. And this is also when we looked at one of the main targets of the heat shock factor, HSP 16.2, we didn't really see any gene expression changes in ASH2 and Z2 depletion strains. We didn't see any more protein to be made. So we could exclude that whatever we see and the positive effect of HSF1 is most likely not through the upregulation of heat shock genes. So, but what then? So turns out that heat shock factor 1, and this is also only been known since about 5 or 6 years, that it also controls many, many heat shock independent targets. And that's often particularly through development. And one of the most upregulated genes of HSF1 turns out to be FET7, which is a desaturase that controls the lipid homeostasis in worms, particularly by accumulating oleic acid. So we thought, well, and this was interesting because a previous work suggested that these monounsaturated fatty acid link the H3K4 trimethylation modifiers to C. elegans lifespan. And so then our model now was, is it possible that HSF1 controls FET7 and through that causes the resistance to proteotoxicity? And as you see here, it looks very promising since the ASH2 and Z2 all have much higher levels of FET7 mRNA. And this is fully HSF1 dependent. And we see this also when we express a FET7 sensor, that they are that these two strains have much higher levels of the FET7 protein compared to HSF1. So now we can put this the next step in our model, now that, and this is something that hasn't been known before, that HSF1 is regulated by H3K4 trimethylation, which then increases the level of FET7. And FET7 is absolutely necessary for the protective effect of the ASH2, because when we take a FET7 depletion strain, and that's in yellow, which is significantly more sensitive than the wild type in black, when we in this background now deplete ASH2 or Z2, we again do not see any more beneficial effect. So this tells us that ASH2 works through HSF1, which works through FET7. Now, in the last 2 slides, I just want to show you, we've had this idea, or we showed you that much of what happens is defined actually in the L2 larval stage, where we see this increase in ROS levels. And so the question is, are any of those participants in this pathway also only really important for during development, and then they just persist or have longitudinal effects? And that seems to be the case because FET7 gets specifically upregulated upon expression of the paraquat in the L2 larval stage. So that fits very well to that. And when we deplete FET7 only during the development, and either the entire time or just during the development, it has already detrimental effects. So it suggests that whatever happens during development affects the heat shock response, it affects the heat shock factor and affects the FET7 activity. And how this persists then throughout life is something that we are very interested in now. So what I showed you is, and this is fits very nicely to what Cynthia Kenyon, one of the leaders in the aging field, said a few years ago. It is possible that a stochastic event, a metabolic insult or a noise in the expression of a regulatory gene, flips an epigenetic switch and or sets in motion a chain of events that promotes aging. So I hope that I convinced you today, or I hope that I inspired you today that this is actually the case, that we found redox variations within a population to trigger this epigenetic switch by downregulating and modulating the activity of the compass complex, which then affects the deposition of histone 3K4 trimethylation. And those that have lower levels of HDK4 trimethylation have an improved heat shock factor response, an improved in the higher levels of FET7. And all these variations only seem to be necessary during the development, but then their consequences persist throughout the entire life. With that, I would like to thank the people who did the work. Brendan, I already acknowledged. Janak is a graduate student who's working on other epigenetic and histone modifiers that are redox sensitive. Agnes works on the stem cell project, and we have former members, Daphne, who did most of the work on sorting the worms and finding HDK4 trimethylation as a redox sensitive mark. I would like to thank my funding, and particularly of course, also my collaborators, and particularly you who all listened to me, and I'm happy to later take questions. Thank you very much.
Thank you so much. Fascinating talk. I have a lot of questions, but like I said, we'll delay them until we have the panel discussion. Thank you so much. We're going to give you a virtual applause again.
Thank you. Thank you.
Thank you. So the last talk of the morning session is by Grey Monroe from the UC Davis. And so we've just talked about longevity and aging, right? Something most of us can relate to. And Grey has built his career around another very, very critical aspect of life, which is the genomic adaptation and evolution in response to climate changes, especially in plants. Grey directs the lab focused on genomics of climate. He directs the genomics of climate adaptation lab, which studies the causes and consequences of climate changes in plants. Plants are in particular interesting because they are extremely plastic, which makes it, I think, a fascinating model to explore the role of mutations in evolution with clear practical implications, such as addressing climate change or consequences of climate change. Grey has also received multiple awards and recognition and accolades for his groundbreaking work. Grey, we're very happy to have you here. I think it's a wonderful compliment to all the other talks. So without further ado, I would give you the virtual microphone.
All right. Yeah, thanks so much. Thanks for inviting me to be here. Let's make sure you guys are seeing my screen. Everything good? You guys are seeing the shared screen, correct? The presentation?
Yes.
Okay, great. All right. So hi, everyone. I'm really enjoying these talks this morning. So I come from a background in evolutionary biology, and it's fascinating to see all this amazing work on sort of understanding biology from a physical perspective. And we've been doing some recent work studying the mutation rate differences across plant genomes. And it's become increasingly clear to me that really taking a biophysical perspective is critical if we want to understand how targeted DNA repair can work, how certain regions of the genome can be preferentially protected from mutation. We have to sort of combine both evolutionary and biophysical mechanistic perspectives to really understand what's going on. So I'm excited to tell you about work that we've been doing in this area in plants. I'll start off by thanking people who've contributed to the work I'll talk about. So we recently published a paper with a large group of people here, and we received great feedback from a lot of folks on this, our funding and our new lab. So we actually just started our lab about a year ago at UC Davis. Before that, I was a postdoc at the Max Planck Institute for Developmental Biology, but we're now in Davis, and we're really excited about continuing to work on questions about mutation and adaptation in plants. So as an evolutionary biologist, of course, well, I guess as a biologist in general, we're of course fascinated by diversity that we see in species across the tree of life. But what's so interesting about this diversity is that it all starts with a rather humble beginning, just beginning by a simple mutation. So all of the heritable diversity that we observe in nature and in humans is ultimately caused by some genetic mutation that occurred, where a change in the DNA sequence from one generation to the next. So understanding the mechanisms that govern why some mutations happen more than others has important implications for understanding evolution and diversity. So many of you, maybe you've heard this, in your kind of intro evolutionary textbooks, this idea that mutations are random, this concept that mutations occur independently with respect to their consequences. So this is one of the oldest ideas in modern evolutionary biology, originating about a century ago during the modern synthesis. And so we can see this kind of summarized here, that mutations are random because they occur independently of whether they're beneficial or harmful. And so this sort of theory, this idea has been defended more recently by explicit theoretical assertions of this idea that non-random mutations, or something that deviates from this, is theoretically untenable because of a lack of mechanisms that can simultaneously tune the mutabilities of multiple genes with similar fractions of deleterious mutations. What this basically means is that non-random mutational processes, if we sort of put on from an evolutionary perspective, are questionable because selection has is very short-sighted. So a change in mutation rate in one gene is unlikely to be appreciable in its selective effect to be even seen by selection. So you can imagine, if the price of milk increases by 1 cent, it's probably not going to affect your decision about whether you buy it or not. However, if you are a cheese manufacturer and you're buying hundreds of thousands of gallons of milk, then perhaps the price of 1 cent starts to matter. So this leverage of whether you can changing mutabilities of multiple genes would if there are mechanisms that can do that, that would satisfy this theoretical challenge that's been proposed to the idea of non-random mutation. So this is the sort of theoretical backdrop on which we'll talk about the work that we'll discuss today, and we'll see that this is being challenged by a number of discoveries across the tree of life. So in the last few years, there's been a number of it's become clear that mutation rates are distributed in putatively non-random ways across the genome. So in E. coli, it's been shown that mutation rates are lower inside of coding regions relative to non-coding regions across a number of independent experiments. There's been excellent work in humans where we know that, especially in somatic mutation distributions, the mutation rate is lower in exons versus intergenic regions, and we can see that across a number of tissue types. And also genes that are highly expressed tend to have lower mutation rates than genes that are lowly expressed. So genes that are silent mutate more often than genes that are expressed more. So this is sort of moving, we're starting to move away from some ideas about mutational randomness perhaps here. And then we also see this in other species. So in C. elegans and others, we see this repeated pattern of reduced mutation rates in exons relative to non-coding regions. So clearly there's something going on here. And today we're going to talk about a work that's happening in plants. So what we know, what we knew is that there's evidence that mutation rates are lower in coding regions of plants, or genic regions, so including coding regions and introns. So in wild type plants, we see a reduction of we see a lower mutation rate in coding regions relative to the expectation of their proportion of the genome. However, in lines that lack mismatch repair, in this case, it's specifically MSH2, and we'll talk about why that's important in a little bit. They have an increase in relative mutation rate in coding regions and other genic features. So clearly there's something going on here about DNA mismatch repair being able to recognize gene bodies and preferentially reduce mutation rates in them. So how does this work? So how, I mean, so as an evolutionary biologist, this seems somewhat problematic given that theoretical prediction that these kind of patterns should be untenable, because what is a plausible mechanism for how mutation rates can be preferentially reduced in regions like coding regions throughout the genome? So how can that possibly work mechanistically? And the key, I think, insight that's emerged in recent years is that the epigenome is a scaffold on which there is a rich amount of information about what is the functional relevance of the DNA sequence in and around the genome. So the epigenome, we refer to, in this case, we're including DNA methylation as well as histone modifications. We'll talk mostly today about histone modifications. We just heard some excellent work about H3K4 trimethylation, but today we're going to talk a lot about H3K4 monomethylation as a mark that's important in plant genomes. But this distribution of this non-random distribution of epigenomic marks provides the information for mechanisms to evolve to take advantage of that information to preferentially repair some regions of the genome over others. And this idea that the epigenome is facilitating DNA repair, is providing some information, is confirmed. This has been well established, especially in the realm of cancer genomics, where there are known relationships between epigenomic features such as histone modifications and things like DNA repair. But today we're going to talk about a story in a different system. We're going to talk about work in plants, and we'll see how it's different than what's happening in animals, which I think is interesting, but also similar at the same time. So this plant here, this is Arabidopsis thaliana. This is a great model system for studying mutation because it has a rather small genome. Its genome is only about 120 megabases. It's inbred, so we have homozygous lines. It has a short generation time, so we can grow it for multiple generations and then count mutations at the end of that process. It's a well-studied model system with lots of genetic tools for knockouts and so forth. So this is the system that we study in our lab as a tool for dissecting the mutation rate differences across the genome and the mechanisms underlying that. So the experiments that this involves to study mutation rates in this species are honestly they're almost boring, because basically we are just allowing mutations to accumulate over multiple generations. So we begin by sequencing the genome of a founder line. And then over many generations, we cultivate by single seed descent. And this is important because we only choose one seed per generation, which randomly choosing that seed, which effectively removes natural selection, because when population size equals 1, there's no opportunity for selection to really occur, except for the most lethal of mutations. But we do this for many generations and then sequence at the end of this process, and we can observe what mutations have arisen. So I'll show data now about 107 replicates of such a process that were each propagated for 25 generations. So we measure germline mutations that were inherited by the final generation over 25 generations. We also added some additional analyses to try to identify putative somatic mutations that were in the leaf tissue of this final generation. And this gives us a nice number of mutations that we can then use to ask where do mutations occur across the genome of Arabidopsis. So when we do this, we find signals of that epigenomic features predict differences in mutation rate, both for single nucleotide variants and for insertions and deletions. So we see a consistent reduction of mutation in GC content associated with GC content, and this is predicted by the fact that high GC regions have a low rate of DNA denaturation. But importantly, we also see a consistent association of low mutation rates, both for single nucleotide variants and indels, associated with regions that have higher levels of this histone modification, H3K4 monomethylation. So, so this provides a sort of a potential mechanism to explain why we might see differential mutation rates across the Arabidopsis genome. And so we've been now exploring this more explicitly. So with collaborators at the University of Tokyo, we've grown knockout lines that lack H3K4 monomethylation. So these are 3 genes that are known to be responsible for establishing this mark in plants. And the triple knockout has a severe depletion of H3K4 monomethylation. One interesting fact about this mark in plants, and this is actually different than in animals and other species, is that H3K4 monomethylation is the most highly enriched mark in coding regions of any epigenomic feature that's been studied. And so this provides an opportunity for us to test the hypothesis that in the absence of H3K4 monomethylation, do we see an increase in the mutation rate, the relative mutation rate in coding regions relative to the rest of the genome? And when we've done these experiments and we see about a doubling of the coding region mutation rate in these lines that lack H3K4 or are depleted for H3K4 monomethylation. So basically, in this triple knockout, we see approximately a one-fold increase in mutation rate of coding regions, and this would be consistent with them being specifically enriched inside of coding sequences. So this raises the question. So these marks are enriched in coding regions. We see when they're lacking, we see an increase in mutation rates in coding regions. But how is this actually mediated? What is the process? What is the actual mechanism by which H3K4 monomethylation is targeted? So how could DNA repair preferentially recognize this mark and then use it as a signal to increase repair rates and thus reduce mutation rates in these regions? So fortunately, there's been some great work on this gene, RDM15, also called PDS5C. This is a gene that does a number of different things, but what's important for us to note now is that it has a specific protein binding domain. It's called the tutor domain. It specifically recognizes H3K4 monomethylation. So in this nice paper, this group did. They did the crystallography, they did the calorimetric measurements to confirm that this tutor domain of this gene is preferentially binding H3K4 monomethylation. It does have some binding of di and trimethylation, but it's much stronger for monomethylation. And they also did experiments where we see where they basically swap out amino acids in the critical binding site. So this tutor domain has an aromatic cage which recognizes the monomethylation. So that's what this is. This is the monomethylation of the histone tail shown here, and this is the result of crystallography. So we know the key amino acids in this aromatic cage responsible for this binding, and it also binds a neighboring amino acid in the histone tail, which gives it the specificity to the 4th lysine. But this provides a really nice way for us to then ask, are genes that have tutor domains doing things related to DNA repair? And it turns out that actually this gene is already known to be involved in DNA repair. In this case, it's involved. It appears to be involved in homology-directed repair. So during meiosis and during somatic recombination, we see it's contributing to DNA repair via homology-directed repair. And this is some great work done by Monica Pradillo. So this is a great example of something that can be potentially binding H3K4 monomethylation, increasing repair in these regions. We can then ask what other genes have this tutor domain. We can look across the Arabidopsis genome and see what other genes have the ability to bind this histone modification. The first thing we see is the orthologs of this PDS5 C gene, and we find one more gene that has this, and this is mismatch repair gene 6. So this is the predicted structure of MSH6 in Arabidopsis. So it has its repair domain here, and then it has a long kind of unstructured tail that's several hundred amino acids long that kind of terminates with this tutor domain at the end. And so it's actually kind of a rather elegant model, physical model, where you can imagine that you have this tutor domain, which is binding H3K4 monomethylation, providing kind of an anchor to these regions of the genome, which is tethered by this long tail, and then it gives room for MSH6 to then sort of search and repair in a kind of regional sense in the neighborhood of these features. So we've been doing work to try to confirm that the MSH6 tutor domain is actually truly binding H3K4 monomethylation. We've started with in silico predictions. So this is the tutor domain of this RDM15 PDS5 gene that's been confirmed via crystallography to participate in this binding. When we look at when we put the tutor domain of MSH6 in Rosetta prediction models, we see that it's predicted to satisfy the same folding structures and relationships between H3K4 monomethylation and its aromatic cage, and its ability to bind the neighboring amino acid. And so this is great work that's done in collaboration with Diego Mateos, who's in the biophysics department here at UC Davis. So we are shown here is a visual of MSH6 and RDM15 tutor domains overlapping each other. This is done by PhD student Daniela Quiraz, where we can see that this here is the H3K4 monomethylation, and we can see that the aromatic cage is fitting a monomethylation. In the same way that it's been confirmed by crystallography in RDM15. So we've been trying to confirm these predictions. We've done in silico predictions. We're now doing in vitro and in vivo models, but here's the in silico results. So we can see that using Rosetta protein-protein binding interaction predictions, we can see that the relationship between the ability for the MSH6 tutor domain to bind H3K4 is highest in the monomethylated state. And this would be consistent with the calorimetric measurements that were made in RDM15. So to kind of visualize that, we have the 4 different scenarios. So in the absence of any methyl group, the H3K4 basically doesn't have anything to enter this aromatic pocket. If we have dimethylation or trimethylation, it basically just doesn't fit inside of the pocket. It's only when we have monomethylation that we see this nice that it fits nicely into this pocket based on the amino acids that are found therein. So I think this is sort of a nice reminder, I guess, for our group as we think about the possible mechanisms of how mutation rates are dictated across the genome, that it can really come down to these very precise biophysical interactions between something like the number of methyl groups on a lysine and the specific binding pocket of the DNA repair gene. And we can actually start doing models where we swap out amino acids So we can look for other genes that are predicted to bind H3K4 trimethylation, for example, and by swapping a few key amino acids. So this is the structure of the MSH6 tutor domain, and if we change a few key amino acids, we can see that then in silico predictions show that it's predicted to bind H3K4 trimethylation. So it really shows the sensitivity of these binding domains to the key residue changes, to their ability to recognize different histone modifications. So we're interested to understand the evolutionary history of this domain, the tutor domain in MSH6. So this is a really cartoony sort of preliminary tree of life here, where we've scanned across genomes and asked, where do we see the tutor domain in MSH6 across the tree of life? And what we find is that the tutor domain, which is this little blue symbol here, is found exclusively in the lineage of eukaryotes that gives rise to plants and other photosynthetic organisms, something around 1 billion years ago. So this is actually really old. And it's also really interesting because it's different than what we see in animals, for example. So it's actually known, and some of you may be quite familiar with this, that in humans, for example, MSH6 has a PWWP domain, which targets H3K36 trimethylation. And this has been established and proves to be quite important for understanding the dynamics of cancer, for example. But what's really interesting and quite compelling about this is that H3K36 trimethylation is the mark of gene bodies in actively expressed genes in humans, whereas in plants, it's H3K4 monomethylation that marks gene bodies in actively expressed genes. So it appears that there's this really compelling example of convergent evolution, where across these really diverged lineages, you have convergence on the same functional outcome, where you have mismatch repair preferentially binding and recognizing the histone modification that marks gene bodies in these super diverged lineages. So I think that's actually a really cool example of a convergence. When we zoom in on only land plants, which we have a lot of genomic information about, we can look at the tutor domain sequence and look for conservation of amino acids. And what we find is that the amino acids that are known to be, that have been confirmed experimentally to be required for H3K4 monomethylation binding, these are the ones that are the most conserved. So if we visualize that on the actual structure, what we see is that these blue amino acids are the ones that we see the highest conservation across. This is about 500 million years of evolution. What we see is that those conserved amino acids are the same ones that are all of the amino acids responsible for H3K4 monomethylation binding are among the most conserved amino acids. And so this suggests there appears to be really strong selection on the ability for MSH6 to have this tutor domain that specifically binds H3K4 monomethylation. So how is this working? So I want to just quickly return to this result that I talked about at the beginning. We have this MSH2 result where in MSH2 knockout lines we see that they increase mutation rate in gene bodies, but MSH2 does not have a tutor domain. So how is this working? Well, of course, maybe many of you know this, but we know that MSH2 and MSH6 operate as a dimer, and so it appears that it's likely that this relationship, the ability for MSH2 to preferentially repair gene bodies, is possibly mediated by its operating in conjunction with MSH6, which has this tutor domain. So basically, the simple mechanistic model of how these repair can be targeted in plants is our hypothesis is that we have our histone modification, and this is being recognized by the tutor domain of MSH6. It has a long tail connecting it to the repair complex which is then free to sort of explore the neighborhood and repair genes in a sort of targeted fashion, depending on the location of where H3K4 monomethylation is distributed across the genome. So this is our basic mechanistic and sort of evolutionary model, that we have the origin of this tutor domain in MSH6, possibly 1 billion years ago. We're still kind of figuring that out. That appears to be able to bind and preferentially target H3K4 monomethylation. And then there's also these other PDS5 genes that I think are doing interesting things as well that we're also exploring. So what is the evolutionary benefit of this? I kind of showed that there appears to be really strong natural selection on preserving the ability for tutor domain to bind H3K4 monomethylation. So there must be some evolutionary advantage of having this. That's what it suggests. And so we can start asking questions about what that might be. So when we look at the sort of the anatomy of a gene in plants, which is different than animals. And so this is important sort of note, is that in plants, H3K4 monomethylation is basically, this is the gene body mark. It's the mark that's found almost exclusively inside of gene bodies. And it's highly predicted to be found in genes that are actively expressed. So this is, from an evolutionary perspective, quite important because gene bodies obviously contain the coding sequences that we know are under elevated purifying selection. So if we look across the Arabidopsis genome and do sort of evolutionary analyses, we can see that it's the coding regions that are under the strongest purifying selection. So these are the most constrained regions of the genome, and so it's not sort of a stretch of the imagination to see how having the enrichment of these features inside of coding regions provides the benefit of reducing mutation rates in these places of the genome that otherwise would be deleterious. So we can actually use the epigenome to just make predictions about what the predicted mutation rate is in and around genes. So here we're showing the predicted mutation rate around transcription start and termination sites. I'll just point out that these rising curves are a reflection of the distribution of gene lengths and not that the mutation rate is predicted to increase deep inside of gene bodies. This is just showing the fact that not all genes in the Arabidopsis genome actually are this long. So we're actually, as we move deeper in genes, we're really just starting to sample onto the other side of gene bodies. But what we can see is that there's this prediction of this pattern of lower mutation rates in gene bodies and higher in these upstream and downstream regions. We've looked at additional mutation accumulation experiments, and we find the pattern seems to be the predicted pattern by the epigenome appears to be apparent. So we looked at an additional mutation accumulation experiment with about 400 lines, and we see this pattern that's predicted by the epigenome. We also looked at de novo mutations in bottlenecked Arabidopsis, and we see the same thing. When we look at natural populations of Arabidopsis, we see signals of this mutation bias having an effect on evolution. So this is rates of natural polymorphism in Arabidopsis, and we see this signal that's very similar to what's predicted, the predicted mutation rate based on epigenomic features. And then we can also use this metric to G-D to distinguish whether this pattern is driven purely by selection or whether this really is mutation bias. And the fact that we see this inversion, the inverted relationship between polymorphism and D is indicative of the signature of what we would expect if this signal is actually driven largely by mutation rate differences and not entirely by selection on random mutation. We see similar patterns in a number of different plant species. So as I said, these mechanisms appear to be highly conserved across plants. So we see a kind of a general trend of this reduced patterns of polymorphism. So this is natural polymorphism in different plant species. However, there's also differences. We see differences between these different species. And so this raises some really interesting questions about what are the underlying causes of that as well. So that's something we're also actively exploring, is looking at sort of where these patterns are similar between species and also where they're different. We also see interesting relationships between mutation rate and the sort of architecture of gene structure. So when we look at genes that have different numbers of exons, what we find is that genes with many exons. So these are all the genes that have 14 exons, for example. What we see is that there's this really distinctive predicted U-shaped curve of mutation rates, where we expect to see low mutation rates deep inside in the exons that are deep inside of genes, and high mutation rates on the sort of peripheral exons. And this pattern is manifest in rates of polymorphism in natural populations. So we see this kind of U-shape of sort of rates of sequence evolution, which appears to reflect this distribution of mutation rates. What's one interesting emergent phenomenon is that it means that genes that have more introns have lower predicted mutation rates and lower polymorphism. And that's basically because having more introns means there's more proportion of coding sequence that's in this kind of low mutation rate zone that's deep inside of gene bodies. And this pattern appears to be consistent with the role of H3K4 monomethylation, which is known to be, which we found to be highly enriched inside of this sort of deep inside of gene bodies, and it depleted at the peripheral exons. And so having more introns means you have more coding sequence that's in this deep this state of elevated H3K4 monomethylation. Which raises some cool questions about the sort of evolutionary relevance of introns in plants. So we also find that H3K4 monomethylation is enriched inside of essential genes. So these are genes that have been identified by knockout experiments in Arabidopsis. We know that they're particularly important for sort of core biological functions, and they tend to be enriched for this feature. So it suggests that they're getting preferential repair. When we look at rates of evolution in genes as a function of H3K4 monomethylation, we find that genes that have very little H3K4 monomethylation are evolving much faster. These are synonymous polymorphisms per base pair. And so these are changes that don't affect amino acid and protein coding regions, but they are mutations, they are polymorphisms therein. They are enriched in genes that lack H3K4 monomethylation and depleted in genes that have lots of this mark. We also see similar results when we look at non-synonymous polymorphism. So there's a strong enrichment of non-synonymous variation in genes that have very little H3K4 monomethylation. We see the similar patterns when we look at divergence. So this is differences between species. We see an enrichment of variation between species as a function of levels of H3K4 monomethylation. And then very importantly, we find that the ratio of non-synonymous to synonymous variation, both polymorphic and divergence between species, is highly predicted by the rate of H3K4 monomethylation. And what this means is that the genes that are under the strongest evolutionary constraint, which we can estimate by this ratio, are the ones that have the most H3K4 monomethylation. And this turns out, this is very important because now we're able to sort of start to satisfy some of these problems, these theoretical problems that have been proposed, that non-random mutation is untenable in the lack of mechanisms that can simultaneously tune the mutabilities of multiple genes with similar fractions of deleterious mutations. But here, the ability for H3K4 monomethylation to be targeted for repair satisfies this problem, because it means that genes that are under similar fractions of deleterious mutations, both differences in genes, and also we showed before that coding regions in general tend to be targeted. And so this provides sort of a resolution to this theoretical problem. So kind of to just wrap it up, so what we're finding is that the mechanisms of targeted repair, which appear to be conceptually consistent but mechanistically independent between animals and plants, is that we have epigenome regulators that are depositing certain histone modifications in non-random ways in and around genes. These become targets for the evolution of relationships where DNA repair is preferentially targeting these regions. And this is also giving rise to differences between mutation rates in genes that are distinguishable by their function and their evolutionary relevance. So essential housekeeping genes, for example, are enriched for features that then can serve as targets for elevated repair. And so with that, I'll kind of leave us with some nice thoughts. So I love this quote that nothing in biology makes sense except in the light of evolution. But I think we might possibly think about amending it to also consider that nothing in evolution makes sense except in the light of biology, and this is especially true, I think, for genome evolution and genome biology. And so, in the spirit of this conference, this symposium, I'll add that maybe nothing in genome evolution makes sense except in the light of genome biophysics. And so we can see that these really precise interactions between just a few amino acids can dictate these big evolutionary processes, like which regions of the genome have mutation more than others. And so I'll just leave it with that, and thank you all for your time. Looking forward to the conversations to come.
Thank you, Grace, so much. So we're going to give you virtual applause too. Thank you. And by the way, when I saw your paper Nature come out, I think in January, I was actually teaching a class in physiology, and I immediately announced the paper, and actually had an interesting discussion. I said I felt like it was one of the most significant new developments in the evolutionary biology.
I appreciate that. Yeah, I mean, I think it's been a long time coming, because I think that, like I said, the cancer genomics community has been kind of ahead of the evolutionary community. I'm kind of catching up on a lot of these things, because I think that these mechanisms are really interesting, and the evolutionary relevance of it has yet to be sort of synthesized, but it's definitely there. But thank you, I appreciate it.
So now we are a little behind the schedule, and we do have another talk at 1 o'clock. So here's what I would like to propose. Perhaps we use a little bit of time, and I promise we're going to take a break shortly, right before 1 o'clock. So in the meantime, what I would like to propose is, why don't we just use the few minutes right now as a Q&A, and we'll just take questions for Grey or Ursula. And we'll go from there. So if you have any questions, please feel free to type them in the chat, either directly to me or share them with everybody. And maybe I'll start with a quick question for Ursula. So let me just make one quick comment. Listening to your talk, I couldn't help myself but thinking about the Hindu concept of karma, and not the karma in the Western corrupted kind of sense, which was confused with the predestination, but more along the lines of that almost everything we do from the time we are born, or perhaps even before that, really impacts the rest of our lives, which is very interesting, kind of merging some Western biology and the Eastern philosophy. Very interesting. One quick question that I had is, so what do you think? Do you think the let's say the mitochondrial haplogroups or mitochondrial DNA, they play a role in those processes? For example, coupling and coupling.
Yeah, I mean, I think that's an excellent question. We haven't done anything in this regard. We haven't started to look at that. I mean, so I cannot really answer this question. We do know that reactive oxygen species, one of the main sources are the mitochondria. I mean, there's no question about that, and how gene expressions there and the whole electron transport chain, how this is affected, we don't know. What is interesting, and I think this is a point that you also made, is we see that early life events can positively affect organisms' lifespan. There are precedents also found in mammalian. So, for instance, in mice, what you can do is you could just deprive them from a little bit of their food in the first 3 weeks after birth, and that would be sufficient to extend the lifespan of those mice by about 15%. Only 3 weeks of a little bit of stress after birth is enough to actually make them much fitter for the rest of the time. Again, to what extent mitochondria play a role, to whatever comes into this. I mean, that's something that we are very, very interested in, but it's very little is known. But I'm convinced that epigenetic changes or histomodifications play a role in that process as well.
Very interesting. And I have one quick question to Grey as well. So I think I feel that this concept that epigenetic modifications may drive the rate of mutations, it's one of those things that, for a while may seem really against the dogma of some of the concepts of evolutionary biology, but when you hear this, it actually makes sense. You feel this, okay, that, of course, it actually does make perfect sense. If you were to design the system anew, that's what you probably would do, right? But I guess the question is, if mutations or the rate of mutations is driven in part by these epigenetic marks, what determines how these marks evolve in the first place?
I think that's a great question. I think that kind of gets to Adam's question about, his question is about, we often think about histone modifications as coordinating expression, but we should be thinking about them more as protection from mutation. And so which came first? It's sort of like a chicken and egg kind of question here about, where did this come from? I mean, I think if we look at, one, it might be kind of difficult to determine because this stuff is so ancient. It appears to be going back, 1 billion years. It's like, this is at the very origin of eukaryotes. This is perhaps one of the key innovations of eukaryotic, success is that they have the ability to have complex chromatin that has these epigenetic features that can then be used to inform things like DNA repair. But I want to, emphasize, I totally agree with your point about how, this seems kind of counterintuitive to some of the ideas about mutational randomness, but when you actually think about it, it makes total sense from an evolutionary perspective, because it's a benefit to be able to, there's a selective advantage to being able to preferentially reduce mutation rates in regions of the genome that would cause harm, so it makes sense. I think, a lot of it, this is where I think getting, understanding the mechanisms and the biophysics and the biology of the epigenome is critical for making sense of it from an evolutionary perspective, because all these ideas about mutational randomness, and we have to remember, these were solidified into the, the sort of dogma in like the 1940s, before anybody knew anything about this stuff, so only in the last few, the amount of information about the epigenome and about mechanisms of repair, this stuff has only been really exploding in the last, decade or so. So I just think it's sort of, it's a great example of why the mechanism matters in evolution, I think. But as for like how it evolved, there's two parts happening, right? So you have DNA repair looking for these features, and then you have their distribution across the genome. So is it, is their distribution determined first and then repair happens? Or is there, there may be mechanisms where DNA repair has ability to target features so that it can stay close to the DNA, and then maybe those features get distributed in interesting ways across the genome. But it's a great question. It'll be fun to determine for sure.
So great, and thank you so much. And then we have a question from Rania Berg. Would you like to voice your question?
Sure. Hi, Ursula. Really interesting talk. I was wondering if you'd had a chance to profile whether these changes in H3K4 trimethylation are happening in specific regions of the genome or just kind of generally.
That's a great question. So, people have already looked at H3K4 trimethylation and distribution in the knockdowns. So, and compared this with RNA-seq. We would be very interested, and I think it gets easier to do this for our oxidized and reduced subpopulation. That was something that we've been planning for a long time. The issue is that you need still quite a bit of material to do those experiments. And so sorting the worms, although this sounded like also simple, but it was actually the hardest thing, and took us 5 years to develop how to live sort worms based on a ratiometric sensor. And so we get too few L2 larvae to be able right now to profile the H3K4 trimethylation and where it is. So we can do RNA-seq on that. And even that, So this is our limitations right now, but we're working on that because, of course, we would be very interested, particularly, and we're doing those experiments to see whether the connection with HSF1 might be through changes in H3K4 trimethylation marks in the HSF1 upstream region. So that would be one of the ideas. But yes, these are excellent. It's an excellent idea, which it's just technically fairly challenging.
That makes a lot of sense. Thanks.
Thank you. And then there is one more question from Adam Williams.
So I can read the question.
Okay, I can read the question. Is the Ross effect transgenerational? Right. So let's start with that.
Yeah. So H3K4 trimethylation is known to be a transgenerational mark. And we see in the elegance that the effects are transgenerational. And Brune has shown that as well when she depletes H3K4 trimethylation. So yeah, so I can tell you that. I don't, what else can I tell you? We've tried to see whether so we sorted the worms and then took the oxidized and reduced and grew them up and got their offspring and looked at them. And it seems like that there is some transgenerational effect that the offspring are still more oxidized when they come from the more oxidized mothers. Yes.
Right. This is a fascinating question, and we actually discussed your work a little bit in one of our meetings, and this is actually one of the questions that was quite interesting from a human perspective.
Yeah, but I think this is what we are actually looking at now is, and this is something that I think is very heavily studied now. What is the effect of the maternal state, of the maternal redox state, of the maternal health state on the offspring? And I mean, this is well known that for instance, offspring from older mothers are typically shorter lived. So this is something that people have observed empirically in many different systems and more experimentally in others. So, and we are actively investigating whether there is a change in redox and some talk between the soma and the germline in the mothers that might affect then the redox state of the offspring, which then affects their lifespan. Yes. Right,
and following up on that, when we talk about the role of the mother, are you thinking along the lines? So are we looking at events pre-conception, or are we looking at, well, in mammalian case, let's say intrauterine environment? I mean, obviously, this is not, you don't have intrauterine environment per se in worms, but in mammalian case, would that be?
So what we see is so we can take worms. So I'm just to show you how early this happens. So we can take wild type worms where we have redox sensors in both the soma and the germline. And we take these worms and we cause a 2-hour food starvation, which immediately causes an increase in oxidative stress in the worms. The germline, which is maybe 15, 20 cells at that point, immediately gets reduced as a response, presumably. And it stays reduced for the whole duration of the development, and even then when they are in their L2 larval stage, they are reduced and ultimately they're shorter lived. So that's a worm, of course. We would be super interested to look at this also in a mammalian system. I mean, but that's of course much more difficult, but that's certainly one of the things that we have that we are thinking about ultimately, to see how redox changes in the womb, because we know that all these many different environmental challenges, or just food restriction, oxidative stress is a very very fast response for food restriction in a wild in many different organisms. So we are quite interested in this. Yes.
So wonderful. All right. So we have the next talk at 1 o'clock. Perhaps what I would like to propose is for us to take a short break, maybe have some food or water, and let's reconvene at 1 o'clock. Thank you.
Thank you.
Hi, everybody. It's Cigall calling. Can you hear me?
Hi.
We can hear you. We are on a short
Great.
break right now, and I think we will go ahead with your talk at 1 o'clock.
That's perfect. Just wanted to make sure I was on time.
You're perfectly on time. We just finished with the Q&A, and we're taking a 10-minute break for people just to grab some food.
No problem. That sounds great. I will be. Do you want me to try to share my slides just in case, or no?
Sure, of course.
Let me try that just in case. All right. Can you see? Let's see here. Can you see these?
Yes. Yeah.
Great. And can you see my pointer?
Yes, we see the pointer. Yeah.
Outstanding. Should I just leave it?
Yeah, just leave it. So
Okay. Might as well leave it.
It's working. So, as
Okay.
they say, unless it's Baroque, don't fix it.
Don't fix it if it's not broken. That's right. Exactly right. All right. Well, we know it works. I may do a couple more things on the computer and just check them over. So I will stop sharing, but we know it works. So we're in good shape. And
the original quote is, yes, if it's unless it's broken, don't fix it. But the one I like is, unless it's Baroque, don't fix it.
Oh, even better. Yes, that's good.
It's from the Beauty and the Beast. We have a 9-year-old boy, so we watch a lot of Disney. So
we like those too.
I'm ready to come up with a quote.
That's wonderful. Well, thanks again for having me, and I look forward to the talk. And sorry I couldn't be there for the panel.
No problem at all. All right. So in like 9 minutes, we'll be back. Okay.
Perfect. Okay.
All right, I think we're coming back. Everybody's coming back from
Sure.
short break. All right. I think we might want to begin in just maybe like half a minute. Just.
Oh yeah, no worries. No worries.
People are back.
Here, let me share these slides then.
All right, I think let's begin. Welcome back, everybody. So we have two more fantastic talks. And again, for those of you who just logged in, the logistics is we'll delay the questions. We won't ask questions during the presentations. And then after these two talks are concluded, we'll have a special Q&A, follow-up Q&A session. Welcome back after the break. Hopefully you had a chance to grab a little bit of food or drink. So we're welcome. I'm excited to welcome Cigall Kadoch from Dana-Farber and Harvard Medical School. Cigall had a phenomenal career in epigenetics and is currently recognized as one of the internationally recognized as one of the leading experts in chromatin and gene regulation for the kind of work she received. For this kind of work, she received multiple awards and accolades. Most recently, American Association for Cancer Research Outstanding Achievement in Basic Cancer Research Award, but many others. Cigall is a director of the epigenetics program at the Broad Institute at MIT and Harvard. We are delighted to have you. Thank you so much.
Thank you so much for having me. It's really a great pleasure to be here. I'm so sorry. I'm a little bit under the weather. So if you detect a bit of a cold in my voice, I am just a little bit recovering from that. So I'll try to get through this without coughing too much. But it's really great to be here. And again, thanks for the wonderful invitation for this great symposium. So as some of you may know, our laboratory is interested in understanding the structure and function of large ATP-dependent chromatin remodeling complexes, most notably the mammalian switch SNF, also called BAF family of chromatin remodelers. And we place particular emphasis on understanding how perturbations to the individual components of these large regulators can play driving or contributing roles in human disease. So our lab largely takes a biochemical approach, integrates structural biology, epigenomics, and human genetic technologies, as well as even systems biology and machine learning to really take a full swath of approaches to understand the many angles by which these complexes can malfunction in human disease, and by virtue of studying their malfunction in human disease, be better able to assign their functions in our normal cells and tissues. And so in parallel to that basic work ongoing in my academic lab, I'm fortunate to partner with and participate in groups intensely dedicated toward translating our discoveries and those of others into hopefully meaningful therapeutics for patients. In this regard, I won't present any of their off-label use or investigational use and new data, but I'll just allude to publicly disclosed programs that are being pursued by the company. And these are my other disclosures. So I think this group really needs no introduction, but of course the sequencing of the human genome has just immeasurably advanced our understanding of the genetic underpinnings of human disease. We've seen over the past decade plus now a wave of sequencing studies spanning cancer, neurodevelopmental disorders, immune conditions, cardiac conditions, and many others. And these contexts have really given us a strong and valuable platform upon which to assess how perturbations to chromatin regulatory and many other entities and pathways can play roles in driving disease. And this is one of my favorite examples here, adapted from one of the papers of the suite of cell papers that came out a few years ago now, reporting the fruits of the efforts of a large-scale cancer genome atlas, or the TCGA. So you can see here, this is just one heat map representing essentially all of their efforts, where these groups profiled for exome sequencing over 30 different cancer types. This spanned actually over 10,000 different tumor samples, and they identified 300 driver genes and over 3,000 putative missense driver mutations. So it's really a remarkable effort if you just think about these numbers. And I showed this slide always to reflect the fact that this is a major inspiration for much of the work that we do. So of course you can appreciate here from these from this heat map on the x-axis, these are the various cancer types abbreviated by their TCGA nomenclature, and on the y-axis a number of different signaling and other pathways. So of course we see very frequent mutations in genes that encode members of RTK signaling, PI3 kinase signaling, MAP kinase signaling, many others. These are common culprits and have been for many years now for the drug discovery industry. We also, see mutations in apoptotic regulators, cell cycle regulators, but perhaps the most surprising and unexpected finding to really emerge from these sequencing studies was the idea that chromatin regulatory processes play frequent roles in human disease and are perturbed with very high frequency. So you can see here some of these rows: epigenetic DNA modifiers, histone modifiers. The complex I'm going to tell you about today is so frequently mutated that it's just one complex, one chromatin remodeling complex of a family of many others, but it gets its own row here across all these different cancer types. And we also see very frequent mutations in transcription factors, which of course are known to leverage various facets of the chromatin landscape to elicit their cell, tissue, and ultimately disease-specific phenotypes. So all of these processes in the nucleus for chromatin regulation can be kind of bucketed into these three categories: DNA modifiers, histone modifiers, and ATP-dependent chromatin remodeling complexes. And we know that the genes encoding the members of these various subsets here play roles in dictating whether chromatin is in a heterochromatic conformation, shown here, largely inaccessible to transcriptional machinery, or in this open conformation as shown here, accessible to transcription factors, polymerase, and other transcriptional machinery for active transcription. And it's really the sequence, positioning, and timing of the placement of these types of histone and DNA and chromatin modifiers that play critical roles in directing the development, differentiation, lineage commitment, response to stimuli, and many other important biological processes. And again, as I mentioned, and really strengthened over the past decade of sequencing studies, we now know that defects in the control of this structure represent major causes of human cancer. And so this is a now structure of the large complex that we study, shown here docked on its actual substrate, the nucleosome. Here you can see this is a multi-component ATP-dependent chromatin remodeler, which uses the energy of ATP hydrolysis to essentially move and ratchet DNA nucleosome contacts, thereby enabling accessibility of target sequence DNA. And these are very conserved molecular entities all the way back to yeast, and that's how they get their SWTCHNIF name nomenclature for screens for mate type switching in sucrose fermentation. And then over the years, they've been increasingly studied in Drosophila and ultimately in humans. And even just a few years ago, we really arrived at the full constellation of the subunits found in these complexes. Here's the full list. These are 29 different genes actually that encode the subunits of these regulators, but you can appreciate that there are not 29 subunits in this schematic shown here. And that's because these are actually pieced together in a combinatorial fashion from the products of those 29 genes, giving rise to hundreds of total possibilities by N-choose-R combinatorics for how they can assemble. And we now know that these assemble into three sort of final form entities termed canonical BAF, PBAF, and non-canonical BAF, all within this mammalian SWTCHNIF family. So this diversity is made in part by some mutually exclusive paralog subunits. For example, at one given position or another in the complex, only one of a family of paralog subunits can incorporate. So for example, SMARCA4 and SMARCA2, these are two ATPases, only one in any complex can incorporate. ARID1A and ARID1B is this one right here, and only one can incorporate at a given time. So with that diversity gives rise to the heterogeneity that we see among these complexes. So if we look at this a little bit more and flip this complex here now on its right side up, we can see this is really pieced together from three main modules. There's this catalytic module at the top, this ATPase module, this connecting ART module, and then this very large core module here where the bulk of the specific subunits that demarcate each kind of complex are housed. You can see this is the largest module and gives rise to the bulk of the molecular weight of these complexes. Now, importantly, these C-shaped, Carpenter C-clamp-like shaped complexes grip the nucleosome at these two pockets of high levels of negative charge. This is the H2A, H2B acidic patch regions where two nucleosome subunit contacts come together to create a pocket of negative charge. And as you can imagine, there are pockets of positive charge on these two faces here of the complex that enable the docking properly onto the nucleosome on chromatin. Now, our lab has had a major interest in studying this particular family of chromatin remodeling complexes, owing to the fact, as I mentioned again, from human genetic studies, that these genes encoding the subunits of this family are very frequently mutated in cancer. You can see here, if we just tally now with the modern TCGA, most up-to-date TCGA data across these cancer types, that if we sum and tally the mutation frequencies for each of these genes, this tallies to about 20% of human cancers, which is quite significant. And actually what we see here is different patterns. So ARID1A is actually the most frequently mutated subunit, mutated in about 8% of all cancers. And you can appreciate from this heat map that the pattern of mutation is really quite spread across a number of different tissues derived from a number of cells of origin. However, there are other subunit mutations that are much more punctate in nature across the different cell type and cancer types as shown here. Now, to try to get a better handle on this, our lab has historically focused extensively on some rare cancers. You can appreciate here from the x-axis that a number of these are very well-defined, genomically well-defined cancer types, many of them actually in the pediatric setting, which is why I find my home department at Dana-Farber in pediatric oncology. And you can see that these are mutated. These subunits here encoding the members of these complexes are mutated, in some cases uniformly or near uniformly in these disease subsets. So these provide very strong support for an initiating role. You can see many of these hit the 100% line or come close to hitting that 100% line. Now, some of these key themes that this brings up is that, we have specific subunits mutated in specific cancers, other subunits across a much more wide range, suggesting some defining functional commonalities. And this has given rise to a number of challenges as well as important questions in the field. So if we now again examine this complex, we've really walked our way around, if you will, cherry picking many of those rare disease contexts to try to ascribe function to each of the individual components within this regulator, particularly in the context when some of these subunits are mutated in the context of being deleted. So the subunit is missing from the complex. We've walked our way around the complex trying to understand the impact of those deletions. We've also spent a significant amount of time trying to understand how these are pieced together. These are 11 to 15 or so subunit entities pieced together from the products of those 29 genes. So it's critical to understand the order by which they are assembled. As one can imagine, that deletion of a subunit at the earliest stages of the assembly pathway may wipe out the entire assembly of this complex versus something that happens at the very, very end may just give you a complex with a hole in it. And it was actually these efforts that led us to derive strategies to endogenously purify these complexes, leading to the 3D structure that I show you here. And in parallel, as I mentioned, we try to take as many approaches as we can to understand not only structural and biochemical modularity, but functional modularity. And for that, we were able to leverage large-scale CRISPR and shRNA screening efforts, where we can look at correlations and fitness dropout to actually ascribe different forms of these complexes. As you can see, this actually dictates which of the types of complexes just by fitness similarity alone across hundreds of cancer cell lines. And then finally, we've invested a significant amount of time in trying to understand how these large regulators interface transiently, but importantly, with transcription factors, which can hijack these complexes or guide these complexes to their specific recognition sequences on chromatin. I'm getting a little bit of feedback. I'm not sure if somebody doesn't have it on mute. Okay. So from some of these studies, we've excitingly been able to identify several therapeutically actionable dependencies that can arise from, exactly as I mentioned, these paralog deletions or mutations. Turns out that complexes missing one paralog, often the dominant one, can then be highly dependent on the residual paralog, in this case, SMARCA2 when there's a SMARCA4 deletion, ARID1B when there's an ARID1A deletion, and so on. We see some shifted assembly and activities of the three different complexes upon certain perturbations. As I showed you in some of the rare cancers, there are fusion oncoproteins that can incorporate. And given the transcription factors that can all interact with these complexes, transcription factor addicted cancers tend to be highly sensitive to the depletion of the function of these large regulators. And so taken together, this has really provided also additional context for the tissue-specific nature of some mutations. Of course, the development of new therapeutic approaches, and also understanding with the cell intrinsic work, the impact on the cell extrinsic microenvironment, for example, of tumors. And I'm excited to say that a number of these have now entered phase 1 clinical trials with new agents in human studies. So as I said, it's not just one complex, but this is actually 3 different complexes, each of which are demarcated by the incorporation of very specific subunits, as you can see here, as well as the fact that some of them are missing certain subunits. And this is how we ascribe which complex we're talking about. Again, the majority of the changes, as you can see, are in the core module, as I pointed out before. So they have very different biochemical compositions, 3D structure, and nucleosome interactions. And in addition, as I told you, we've looked at the assembly pathway to understand how these different complexes are assembled. Turns out, and I don't intend for you to memorize what's on this slide, but the key points here are that there's an initial core module for all 3 complexes. There's a critical branch point where you can specify which arm of this pathway one is going down to create CBAF complexes, PBAF, or NCBAF. And then there's this critical ATPase capping event at the end where we see that this ATPase module assembles on and gives this complex its catalytic energy. So why do we care about this? Well, this can inform, of course, the impact of cancer-associated mutations. If we know where these mutations occur on the assembly pathway, we'll better be able to understand the ultimate resulting complex and its function. And this gives us a very diverse repertoire of opportunities for inhibition, again, depending on the subset of subunits that are represented in any disease context. Now, in addition to the biochemical features of these complexes, they also go to different places on the genome, as you can imagine from their subunit componentry. So if we just now look at heat maps, looking at the clustering of different subunits for different complexes, this is for the NC-BAF complex, the PBAF complex, and the canonical BAF complex, you can see that this actually separates them out on chromatin. This is reflected if we just look at a cherry-picked locus, for example, the VEGF-A locus, where you can see different enrichment of different complexes throughout that. And then if we look at different features of the chromatin landscape, active enhancers, prime sites, active promoters, CTCF sites, you can see the distribution for each of these is markedly different. So this suggests that these complexes indeed have differences in how they read and respond to the chromatin landscape, which of course would be expected to impact their function in gene regulation. And so recently we took a stab at trying to get at this in a more direct manner than what had been done previously. So this is what we all look at. We're all used to looking at ChIP-seq tracks, and we can look at the localization of complexes, different complexes, and how those line up with histone marks for different features, enhancers, promoters, active and repressed regions, etc., as we can show you here. But an issue with that is that these have major limitations in that this reflects averages among molecules and among cells, given the types of strategies we're using here. This is never direct, it's correlative at best. And because this is working with a chromatin remodeler, for us, unfortunately, the marks that may have played a critical role in actually bringing that complex to that site are often now on the nucleosomes that have been ejected. And hence it won't show up over to the target sites of the chromatin remodeler. So this is challenging for the field, and that it's very challenging to figure out what aspects of the histone code or the chromatin landscape truly play direct effects in guiding these remodelers to those target regions on chromatin. And then finally, the studies that we've seen in yeast to date are quite limited, or with domains of human proteins, but not purified in the context of the whole complex where you have the allostery and all the structural constraints that may be important for some of these interactions. So for us, this really underscored a major need to understand the direct determinants of targeting activity. And if we were able to do that, this would give us a whole new suite of opportunities for how we may be able to inhibit complexes, their modules, even some specific subunits from their interactions with chromatin. And this would give us an understanding, of course, of the basis for these targeting proclivities. So I'll just show you some recent work that we recently published alongside Tom Muir's lab. This was a tight collaboration with his lab at Princeton, where we leveraged a cleverly designed DNA barcoded library that contained a suite of nucleosomes, 118 different ones with all the marks shown here on this radar plot, each with a different DNA barcode. And we purified each of these types of complexes within the M-switch-NF family, canonical BAF, PBAF, or NC-BAF, and we subjected them to this large-scale screening exercise for remodeling activity throughout the different DNA barcoded nucleosomes. And the key point I want you to take away from this information-rich radar plot is that you can see major differences between the 3 different complex types. So you can see the blue often has very inhibited function by a large number of the nucleosomes represented in the library, NC-BAF a little less so, in fact, much more potentiation, and so on and so forth. So we learned a lot about the rules, if you will, of how these complexes get to where they need to go on chromatin. And as a sanity check, I told you these complexes grip the nucleosome at the acidic patch. So indeed, if we chose nucleosomes as shown here that have mutations that disrupt the integrity of the acidic patch, indeed the remodeling activity of these M-switch chromatin remodelers was markedly reduced, as shown by this heat map and mapped here onto the nucleosome structure. And we were able to do that not only for nucleosome core, but also nucleosome or histone tails rather. You can see here, even along one tail, such as the H3 tail, we see some marks vastly potentiating the remodeling activity of these complexes and others inhibiting it. So this gives us new paths toward understanding how they specifically interact with the chromatin landscape. And in addition to that, we've taken another level to try to understand how they are targeted on chromatin. And this came from our work and observation that these complexes, particularly canonical BAF complexes, are strongly enriched across cell types at transcription factor sites. So for example, here are some of the motifs that are enriched particularly for these complexes. And if we now just look at the localization of BAF complexes across tissues, this is just across cell lines that we've profiled in the lab, you can see that there's a shared set of sites where many complexes are localized irrespective of tissue type. And then there's very specific subsets that are extremely specific for the tissue in question. And so this led us to the hypothesis that perhaps transcription factors in the repertoire of them are guiding this. And you can appreciate here that if we go in and dissect each one of those clusters, indeed, there's strong representation of certain types of factors which guide their positioning. And this was true if we now start to look at the tracks under each of these, where you can see very specific sites for heme, specific sites for kidney, specific sites for ovary, specific sites for prostate, and so on and so forth. You can see very specific targeting patterns throughout this. So much so that if we now overexpress a transcription factor, for example, in a cell, in a mesenchymal stem cell, for example, you can appreciate that the complexes, this is a distribution of where they are on motifs, really skew toward the sites that are rich in the motifs for that factor and are taken away from other sites. And this goes as a function of the expression of the factor one is expressing and the motifs on chromatin that are present near those binding sites. So now what's interesting about the mutations that I told you, there's 20% of human cancers, about 60% of those affect just single residues in the complex. And so it behooves us then to leverage our structural understanding of these complexes to map those mutations across the subunits as we've shown you here. So here is a SMARCA4, SMARCA2 shown in the globular format, the rest in ribbon diagram. And you can really appreciate some key hotspots that are highly recurrent for mutations in the bends and the knees, if you will, throughout the complex, as shown there. And we can play this game throughout the entire complex, shown here, mapping the mutations and mutational frequency throughout the complex to try to identify these key hotspots. And our goal over the, now enabled by these structures and increasingly enabled hopefully by more high resolution structures, is to be able to increasingly bucket those families of mutations in pie graphs such as this. For example, what we learned from this is that only 1% of the mutations are actually catalytic in nature, whereas the majority of these really affect core intersubunit interactions, subunit nucleosome interactions, and affect certain modules of the complex. So the goal of this would be to try to increasingly generate functional classes to provide some foundation for therapeutic stratification as we better understand how we can therapeutically interdict for some of these tumor types that are driven by specific compromise to these different regions. And then finally, I'll tell you just one vignette about how we've done that. This is a recent story from the lab where we actually leverage not only cancer sequencing data, but actually also sequencing data from intellectual disability and autism spectrum individuals. And we can appreciate here that there's a pileup of mutations really exclusively in the C-terminal domain of this one subunit, SMARCB1. So we became very interested in understanding what is the contribution of that one C-terminal domain here, about 50 amino acids or so toward the end of this protein. So we generated complexes containing any of these mutations, actually a related mutation in the other side, N-terminal side of this complex subunit, and wild type. And you can appreciate from this silver stain that there's really no compromise to the biochemical integrity of the complex, so that's not the answer. And with that, since we had fully formed complexes in all conditions, we were able to subject these to remodeling assays. And for those not familiar, these are just nucleosome remodeling assays where DNA is wrapped precisely twice around these nucleosomes, and there's a restriction digest site that's taught with the nucleosome here, and only in the presence of an active chromatin remodeler can that DNA sequence become accessible and the enzyme can cut. So you can see it can cut the DNA. And so all one is doing is measuring the presence of the bottom band over time as a measurement of the modeling activity. And excitingly, we were able to show that all of these coffin serous associated, or these intellectual disability associated mutations resulted in a decrease in the remodeling efficiency here of these complexes. And this is shown here over a time course as well. So what is it so unique about this C-terminal region of SMARCB1? To that end, we asked whether or not this C-terminal region would be sufficient itself to bind the nucleosome, as this is portending remodeling activity. And just to put this in perspective, before structural studies, we did not know that this is the region of the complex, of this multi-component complex that contacts the nucleosome. So we incubated these with nucleosomes, as you can see here, performed biotinylated peptide pull-down experiments. And you can appreciate that the wild type sequence of the C-terminal domain indeed does bind to the nucleosome. However, mutation of that C-terminal region uniformly attenuates the ability of this region to bind and interface with the nucleosome. So all of the coffin serous associated mutations failed to bind the nucleosome. And when we perform HSQC NMR at the time to try to understand this in a more structural manner, you can start to appreciate why that would matter. This is a basic, highly positively charged cluster of amino acids, all on the outward face of an alpha helix, barrel-shaped alpha helix. So you can appreciate how mutation of any of these would be affected, would affect the interaction perhaps with the nucleosome. And we showed using photo cross-linking experiments that this region interface with that highly negatively charged pocket, the H2A, H2B acidic patch is shown here. So much so that if we now try to incubate the wild type C-terminal sequence with nucleosomes that are either wild type or those that have the a problem in the acidic patch, you can see that they only interface with the wild type, and there's markedly reduced engagement with nucleosomes that have a problem or that disrupt the integrity of the acidic patch. This became much more clear, of course, when we had the structure just a few months later, and we could see here that that alpha helix I'm talking about is actually this one right here. And now it makes it much more clear that this is the only site for the entire bottom part of this entire complex to interface with the nucleosome. So without this, it would make sense why the remodeling efficiency would be reduced. You can see that from another angle here. Excitingly, there are a number of cancer-associated mutations that are right at this interface. Look at them. They're all on the outward face of the helix here, all predicted to disrupt this interaction with the H2A, H2B acidic patch. And this was true for the upper side of the complex as well, so much so that if we make these mutations, we can again use these remodeling assays to show diminution in remodeling activities. So that's all in vitro. What happens in cell culture systems? So if we now try to understand this in cells, what is the effect? So if we now take these SMARCB1 deficient cells, this is actually a tumor model, and we introduce either wild type or any of these mutations in the C-terminal region into these cells, and we ask what happens by ChIP-seq. So interestingly, as we perform ChIP-seq for these complexes, you can appreciate that there's actually no difference in the targeting between the wild type or any of these mutants in terms of getting to these sites on chromatin. There's also no difference really in the K27 acetylation recruitment. But when we started to profile the accessibility via ATAC-seq, you can appreciate a marked decrease. It's not gone, but it's very much decreased compared to the wild type for all of these coffin serous intellectual disability associated mutations. You can appreciate that there's a major change, mirroring actually the percent change that we saw in vitro for remodeling defects here compared to the wild type at these de novo sites. You can see that represented here, that the occupancy of these complexes is much the same, but the accessibility peak shown by ATAC-seq are reduced in every case. And if we look at now an IPS model to try to recapitulate the heterozygous nature of the mutations that occur in individuals with coffin serous syndrome, intellectual disability syndrome, you can see that upon neurogenin differentiation of these IPS cells, this is day 8 of stimulation, here's a cluster of genes that are all strongly activated. However, in the context of the heterozygous, one deletion of one lysine residue here, you can see there's a major attenuation in the ability of many of these genes to be activated. And here are some of them shown here, where in every case you see in the gray, the wild type is able to be activated either along the time course or by the 8 days of the time course, but the K364 deletion mutation is unable to be activated to the same extent. So this really suggests a convergence between having an M-switched F master regulatory mutation or an individual developmental pathway associated mutation. So this is one of the ways we've really gone after how to dissect these individual point mutations. And there's a lot more to do. As I mentioned, 60% of these mutations are missense in nature, and they occur in DNA binding domains, histone reader domains, many unstructured regions. So I just reflect this fact to show that we have a lot more work to do to try to decode what is the mechanistic contribution of each of those regions. Along with that, trying to understand how these complexes pair with transcription factors. I've told you, we've looked at how they interface with the histone landscape, combinations of histone marks in the context of cancer and other disease and otherwise, the mutations and marks, for example, phosphorylation marks on these complexes, these unstructured regions, all with the goal and the eye toward understanding the tissue-specific nature of the mutations, new therapeutic opportunities we may have to interact, to interdict the activities of these complexes, and to start taking the bulk of what has been historically very cell intrinsic work to understand for the first time how this may be interplaying with the with the tissue microenvironment or tumor microenvironments in the case of cancer. And so with that, I'll just thank the members of the lab that did this work, my funding sources, and all of you for your attention. Thank you.
Thank you so much. Thank you. So let's give you virtual applause. Thank you. And a reminder to everybody, please feel free to writing down your questions in the chat, either to me or to everybody, and we'll follow up with the questions right after the last talk. So last but not the least, our fifth speaker is Dave Gilbert, from the San Diego Biomedical Research Institute. One of the central themes of Dave's lab currently is understanding replication domains. A very important step and very exciting new direction in research, still very poorly understood. And in fact, a lot of what we now know about the replication domains and their function in the S phase and the relationship to chromatin structure came from Dave's lab. So thank you so much for identifying this very important events. Dave is a recipient of a number of significant awards. I'm not going to mention them because otherwise my introduction would be a little too long. And without further ado, Dave, it's all yours.
Yes, hi. For some reason, I've already had 3 or 4 Zoom meetings this morning. For some reason, it's not allowing me to share. It's telling me to go to System Preferences. So there's something different about our Zoom connection than any others I've had, but I'm going to System Preferences, and Zoom will not be able to record. I don't care about that.
Okay, how about I try to share something to see if...
Let me try now, because I've gone in and done what it asked me to do in System Preferences, and it is now looking like it's going to let me share. Okay, can you see the screen now?
Yes, we can see the screen. Okay,
sorry about that. Didn't anticipate that, but it was the directions from Apple were clear. Great. Thank you very much. I wanted to say that when Vadim and Ben first sent me the invitation, I immediately said yes, because I was intrigued with the concept of physical genomics. And of course, I'm familiar with John Marco's work over many, many years. If he's out there, I'll try not to say how many decades, but pulling on chromosomes and measuring physical forces. And I'm very familiar with Leonid Murney's using physical principles to build 3D models. But I was trying to think of how, my work could be of interest to you guys. And of course, I remember that ever since the 90s, in fact, we've been saying that when replication domains are anchored in early G1, they can then seed the assembly of microenvironments within the nucleus of differing molecular constitution, and that would then set thresholds for replication to be either high potential or lower potential. And of course, we now call that phase separation. We didn't call it that when we first proposed it, but that's a very physical principle. So I decided that I would pull some of those old slides out and tell the students all about what we've learned about replication domains over the years. So in this first slide, I want to illustrate what an exciting opportunity replication domains are for understanding the liaison between omics and imaging, which is a big deal for us in the 4D nucleoma and in the genome architecture field. Is can we validate what we indirectly see by genomics directly by looking at the genome. And so what you see at the top, what we've done is pulse labeled cells briefly with bromodeoxyuridine, and then stained for DNA content and sorted them into 16 fractions of increasing DNA content, so going through S phase, and then expressed a heat map of each fraction. You can see S1 through S16 of the sequences that were replicated during that time. And what you can see is that there are very clear patterns, and they're very deterministic patterns for a biological process. Most of the reads, the vast majority of the reads, are found in a very fine temporal window. At the bottom, you can see what happens if you pulse label cells similarly, but instead fix them, stain them for the label, and then look at them under the microscope. And what you see firstly is that replication takes place when you briefly label. It takes place in these punctate replication foci, just a descriptive word that we still use until we really understand them. And you can see that the spatial patterns in the nucleus change as you go throughout S phase, from the interior of the nucleus to the periphery and around the nucleoli, and then to other sort of mysterious heterochromatin regions that we're still trying to figure out what they are. Okay, so back in the 90s when I started my lab, we didn't have CRISPR and we didn't even have sequenced genomes. And so my lab did a lot of cytology. And so one of the things we could do is to actually look at the dynamics of replication in fixed cells by doing what we call pulse chase pulse experiments. So what we would do, illustrated here, for example, is give a brief pulse with one halogenated nucleotide, iododeoxyuridine, and then chase that label out, and then label later briefly with chlorodeoxyuridine. And then we could look at the DNA that replicated before and after a 4-hour period. And what you see on the right is a cell where in green is the early replicating foci, just as I just showed you in black and white. And then in red are the later replicating foci. And so, you know, I like to say at genome architecture meetings that we had TADs or domains and compartments, early and late replicating compartments, almost 30 years before Hi-C was invented, okay? And we've been thinking about the nucleus in terms of domains and compartments for that long. And I just want to say that Irina Solovey published this nice review article. I don't know if she did this work, I would assume she did, but where she showed that this same pattern is evolutionarily conserved all the way back to Tetrahymena. And yeast don't really show this, but yeast nuclei are about as big as one of these foci. So, you know, Tetrahymena has 100 megabase genome, so it's quite possible you just need a sizable genome. But you can see even the simple eukaryote hydra, multicellular eukaryote hydra, has this pattern of early replication in the interior and late in the periphery, and corresponding to euchromatin and heterochromatin. Okay, so we took advantage of this pulse chase pulse approach to do a few things back quite a long time ago now. And one of the things we could do was measure how long it takes each individual focus to complete replication by doing short chases. And so there we could see, you can see we could go from 100% co-localization of the two labels with a 0 time chase to almost 100%. I've got to move. You guys are in my way. I've got your faces to close you so I can see my own slide. So to almost 100% lack of co-localization after 45 to 60 minutes. Okay, so we conclude that each region, each domain, each segment of DNA takes 45 to 60 minutes to replicate. And then we could do longer time chases to measure the length of each of these patterns by looking for the time it takes for them to transition from one pattern to the next. And we could draw these little cartoons. And we did this for several different cell lines in different species. And we found very, very similar kinetics of the times for each of these patterns. Oops, slide is not advancing. Let me try the zoom advance. Okay, so now another thing we could do, actually a lot of this was pioneered by Cristina Cardoso, but we're currently collaborating with the Allen Institute for Cell Science. And they've made a human iPS cell line that expresses GFP tagged pCNA. So we can track these same replication domains by following the proteins that carry out DNA synthesis. And I'll just show you this movie very quickly. And I picked out 2 cells to show you. This cell here, number 1, is just ending S. It's in the very last pattern, and you can see that it goes into G2 and finishes. And there's a cell over here that's in G1 at the beginning of the movie, and you're going to start to see foci forming. So this is pCNA going to the sites of DNA synthesis. And now we can also label in living cells and track these foci in living cells, that is the DNA, by labeling with fluorescent nucleotides. And these sort of came along later than the halogenated nucleotides, and you have to transfect them in or get them over the membrane somehow, but you can still do it. And so what you can do is label replication past, that is the DNA that replicated during your pulse, and then track that into mitosis, where you can see these cells are in anaphase, into early G1, and then they sort of reform the patterns that resemble the patterns they had in the previous cell cycle, or this is a rather late DNA synthesis pattern. And if we do this in the same cells that express an RFP pCNA, you can see that pCNA is sort of soluble in the nucleus throughout the cell cycle, but it forms these foci during S phase that are very prominent and easy to see. And you can see that when S phase begins, the foci, the labeled DNA is still in the late replicating pattern, and the foci are in an early replicating pattern, so they don't co-localize. And then you can continue to follow these through S and watch pCNA rejoin the DNA that replicated late in the previous S phase. So these are just some of the sort of tools that we can do, and you still use these tools, but of course genomics came along and took over everything. I want to show this other thing you can do is to chase for very long time. So pulse label and then chase for very long times. And what that does is dilutes the label down to just a couple of chromosomes. And that way you can remove a lot of the background noise and really dig into some of these foci. And this was really pushed by Maya Mendoza when he was in Dean Jackson's lab. And Jan Ellenberg has taken advantage of this by using live and super resolution imaging to track these foci labeled exactly as I just said. And he could actually see the sites of label within the domains, but he could show that these domains move as units. So all the DNA within the domain, or all the labeled DNA, is going to move as a unit during the cell cycle, which provided additional evidence that these really are stable chromosome domains, which is sort of the model that my lab had been proposing for a long time. So an experiment we did back in the 90s to try to understand what regulates this timing program, we labeled cells early and late, as I've described, and then chased them into mitosis, where you can see bands on chromosomes. And then we asked 2 questions. One is, how long does it take these patterns to reform after mitosis? And we could do this in living cells as well. But what we found was that in very early G1 phase, now this is after the nuclear envelope has reformed, so there's a nuclear lamina and a membrane around the chromatin. But the chromatin in living cells labeled with fluorescent trinucleotides, we could see that these foci were highly mobile, moving all over the place in the nucleus. And by fixed cell imaging, we could see that the early and late foci was scrambled randomly throughout the nucleus, okay? But then at a time 1 to 2 hours after the nuclear envelope had completed, the nuclear membrane, we could see that these foci then returned to their positions where they would then remain for the rest of interphase, okay? We could then introduce nuclei from these cells into a cell-free system that I developed as a postdoc that could jumpstart S phase within nuclei isolated at any time during G1 phase. So we could ask questions about the preparedness of chromatin for executing the proper replication program, both spatial and origin positions, etc. And what we found was that if we introduced nuclei in these first couple of hours after nuclear envelope formation, replication proceeded in a random temporal order in our cell-free system. And at the time that foci became repositioned, now in that same cell-free system, nuclei, chromatin would replicate in the proper temporal order. So we call that the timing decision point, and this really gave us a lot of confidence that the 3D organization of these foci is important for executing the proper replication timing program. And then many years later, a graduate student, Junji Liu, wanted to determine when this program is lost. We assumed it would be lost at mitosis because that's when the nucleus is blown apart and the positions are lost. But in fact, to our surprise, he found that G2 phase nuclei, either stimulated to initiate replication in our in vitro system or through certain tricks that he used with kinase inhibitors, he could get cells in culture, so live cells, to reinitiate another round of replication. And in both cases, that replication initiated in a random temporal order. So that told us that we're looking for some kind of a element that's assembled early G1 and then is disassembled probably during S phase, and we suspect at the replication fork. Why not? However, we're still looking for these determinants. Now, based on these results, we proposed a rather naive model that as soon as sequences are anchored in the nucleus and stop wiggling around, they can then seed the assembly of microenvironments of differing molecular constitution that could then set thresholds for the initiation of replication, so that some of these subnuclear microenvironments would have low thresholds and would replicate early, and others would have high thresholds and would replicate late. And these are some of the words that we use, and I just like to point this out because now, some people call this liquid-liquid phase separation, but some people call it a seeded nucleation. But it's exactly the same concept. Somehow intramolecular interactions create microenvironments that are not separated by membranes, okay? Okay, so for the next few slides, I'm just going to go to the omics and tell you what we learned about domains from omics. This is our most robust method for looking at replication timing. We label with BRDU, and then we sort only 2 sections of S phase, 2 time points in S phase, basically the first and second half. And then we immunoprecipitate the labeled DNA and sequence it and express it as a log-2 ratio. And this is a really robust method. We can look at dozens of different experimental conditions in one sequencing lane. And this just shows you the history of, I developed this as a graduate student a long time ago. But to talk about this in modern times, if we display in 5 kb bins, the log-2 ratio of the reads in 5 kb bins, you can see that there are indeed these swaths of what we call constant timing regions, large regions that replicate at similar times. And they're punctuated by, sorry, and this is a smooth version of this data. And what I wanted to point out is that these constant timing regions are punctuated by what we call temporal transition regions, where forks are consistent with forks moving unidirectionally for many hours at 2 kb per minute, which is the elongation rate of mammalian DNA synthesis. So we could then segment these domains and ask questions about their sizes, where are their boundaries, etc. And over the years, some of the things that we found is that this program is extremely resistant to all sorts of insults, deleting all kinds of chromatin regulators, treating with chromatin inhibitory drugs like histone deacetylase drugs, etc. This program would just look back at you, and you could barely find any changes. But as soon as we would differentiate either mouse or human embryonic stem cells, we've done a lot of work with both cases, and almost all the concepts are identical between the 2 species. But we would find, as soon as we initiate a cell fate change, we would find a massive reorganization of this program. So this program is developmentally regulated. That was one of our most exciting early findings. And they're coordinated with changes in replication timing, are coordinated with changes in transcription, chromatin, 3D organization of the genome. And particularly what I'm going to talk about now is what we found was that developmental changes occur in units of 400 to 800 kb. And that's when we really started pushing this idea of replication domains. So what we found is if you take this sort of large, multi-megabase constant timing region, if we looked in different cell types, or we differentiated stem cells, these are human stem cells, we'd find that in some cell types this region would be late and early in others, and this region would be late in other cell types, etc. But whenever we saw these changes, they always occurred at approximately the same boundaries within the limits of our resolution, but also they were always in this 400 to 800 kb size. And I want to show you, this was an early finding, as you know, this was our first genome-wide paper. And so in this paper, we only did ESC to NPC, and then later we did tons and tons of cell types. But what we found was that if you look in neural precursors, this is the distribution of the sizes of those constant timing regions. But if we looked at the switching regions, whether they're switching early to late or late to early, they're always in this very tight size range. This is mouse and human differentiation. And so this gave us the first idea that we were really looking at a unit of replication timing regulation during development. And the sizes of these were consistent with they're kind of Carl Sagan-ish sort of estimates of how much DNA must be in a replication foci by cytogenetics, just based on how many foci there are, et cetera. So then after this finding, a postdoc of mine, Shinichiro Takabayashi, who was the best microscopist that I ever had, he wanted to take a look and see if he could visualize these developmental changes. So what he did was he made a bunch of fish probes around one of these domains that contains DPPA genes. These genes are expressed uniquely in pluripotent cells, so stem cells, embryonic stem cells. And this domain that contains these genes is early replicating only in pluripotent cells and switches to late replicating in all other cell types. And this was our favorite differentiation system back then, embryonic stem cells to neural precursors. So he made fish probes that were either within or flanking this domain, and then looked at their relative positions each to each other as we differentiated the cells. And what he found was that in pluripotent cells, this domain was actually quite distended and then compacted upon differentiation. However, the two probes flanking stayed at the same distance as you differentiate. So this domain is kind of open up like a flower when it's in the pluripotent state, and then closes down and joins its neighbors during differentiation. And so this little cartoon we draw is just to illustrate that what we believe is that this domain sort of pops out of its compartment. And I should say we also did 4C, which is an old-fashioned poor person's version of high C. And back in these days when high C was difficult and expensive, we did 4C, which is several baits to the rest of the genome. And what we found was that this domain interacts with the early replicating compartment of the genome in the pluripotent cells and is expanded. And then during differentiation, it joins its neighbors in the late replicating compartment, and now it interacts with its neighbors as well as other late replicating regions. Here, it doesn't interact with its neighbors nearly as well. And the other thing we found, which I'm just going to say, which was a big surprise, was that there was no change in accessibility. We didn't have ATAC-seq back then, but we had micrococcal nuclease sensitivity, DNA sensitivity, and also reviewers asked us to do restriction enzymes. So we did 2 different restriction enzymes of different sizes. And in all cases, there was no change in chromatin accessibility. And the surprise was that developmental domains genome-wide are inaccessible as heterochromatin domains, even when they're early replicating. So this is a different breed of domains that expands but remains inaccessible to attack by nucleases. Very interesting. We still don't understand the structure. That's a physical problem for you. Okay, so then when Hi-C did come out, the very first paper from Job Decker, we noticed right away that when he expressed his Hi-C data as an eigenvector or first principal component of the Hi-C interactions, we saw that these plots looked an awful lot like replication timing plots. So we went and very quickly profiled the same cell line Job used, and we found it looks a lot like replication timing. In fact, the subtle differences in replication timing within the constant timing domains matched subtle differences in interaction frequencies within the compartment regions. So we were wondering whether maybe these bumps here represent the replication domains as self-interacting units. And indeed, as years went on, not too many years, a lot of more high-resolution, high-C data came out, and we also profiled dozens and dozens and dozens of more cell lines and found more and more and more of these boundaries of these developmental domains where we really could say these were units of regulation, and we showed that they align with topologically associating domains, or TADs. So we really started pushing this idea of a replication domain model, that replication domains are TADs, they're stable structures, there's boundaries are preserved in different cell types, but they fold in such a way in different cell types that replication domains that replicate at similar times come together in space in proximity. And the 3D arrangement and replication timing, and we also showed by a lot of 4C that this 3D, that both TADs and compartments are formed at the timing decision point. So this whole 3D arrangement, as well as the replication timing, are formed coincidentally at this time early in G1, a couple hours after nuclear envelope formation. So fast forward to today, we've collaborated with Jennifer Kremens to look at in more detail this relationship between TADs and replication domains. And I'm just going to show you one figure. There's a bioarchive, it's outdated now, but this we hope will be coming out soon. Everybody's familiar with this process. What you're seeing here is a pileup of all the corner dot domains. So all the cohesin extrusion regulated TADs in the genome, all made the same size and all aggregated into one plot. And then what's aligned here is, remember that 16 fraction repli-seq, we piled that up against the, aligned it to this APA plot of the dot domain TADs, and we found that the borders of these TADs align very nicely. It was very enriched with the earliest replication initiation zones. So the earliest sequences to fire, which we believe contain origins for lots and lots of reasons that I don't have time to talk about. But so then we knocked down RAD21 to eliminate loop domains, and we found that very much reduced the enrichment of initiation at the TAD boundaries. And we went on to show this was chromatin loop extrusion, because if we knock out WAPL, I'm just going to tell you in the interest of time, what we find is actually the alignment gets tighter than even wild type. So when you knock out WAPL, that's the cohesin remover. So extrusion is even more robust. And we find that we get even tighter alignment of the origin. So what we propose, just a working model, is that when cohesin is extruding chromatin, it pushes the irreversibly loaded MCM. We showed back in 2011 that MCM is locked down, irreversible, the most tight interaction with DNA of any protein known to man currently. And so that it has to either go through that circle, or it's going to interfere, right, when the two collide. So there is a paper out there in Journal of Biological Chemistry, I think, showing that cohesin in purified systems, cohesin and helicases will bump into each other and push against each other. So that's another physical principle that you might be interested in. So we're proposing that cohesin, when it extrudes, pushes MCM, which is de facto the place where replication initiates. So it pushes the replicative helicase that initiates replication into the boundaries. So that's the model. And of course, we have a lot of work to do to test that model. Okay, so in the last few slides, I'm going to tell you finally a little bit about what regulates replication timing, and then bring our model into up to speed, up to the present day. So this work, the first discovery of cis elements that regulate replication in any way, except in budding yeast, I should say, but I should say in mammalian cells. We can thank this graduate student, she's now a postdoc studying sleep, Zhao Sima. So when Zhao came to me, she said, I want to find the cis elements, and I said, well, good luck. There's hundreds of postdocs and graduate students who have before you who have failed, and in amazing laboratories like Mark Grudin's. But she said, but I have CRISPR. And of course, she was absolutely right. So, I can do high throughput, unbiased deletions. So she did that. And after about 2 years, she had dozens and dozens of deletions. She made eventually over 100. And none of them made any sense. All we got was partial reproducible results. So what we did was we got the highest resolution 3D model that we could get using Capture Hi-C. And when we did that, we found that there were 3 places that were closest together in space and did not have CTCF at both of their points. So they were not CTCF binding sites. And so one of them was, but I mean, there weren't any pairs that were CTCF binding sites. And pairs of CTCF binding sites define cohesin-mediated loops. And we had shown that we can knock out CTCF and it has no effect on replication timing. So we knew we were looking for something that was CTCF independent. So Zhao found these, and she showed me that they also had broad peaks of H3K27 acetylation, which are characteristic of what are called super enhancers. So she said, I'm going to delete those 3. She went in and did that, and when she did it, we got this. This is my career picture that I'll take to my grave. But basically what you can see is 2 replicates, that is 2 independently picked clones with independent deletions of these 3 elements. And you get a complete switch from early replication to late replication, okay? Now, when you have a result like this and people couldn't do this for 60 years, you cannot publish, in my opinion, and if I'm a reviewer, I won't let you publish unless you explain why you did it and nobody else did it after 60 years. And so this was very satisfactory. So what Zhao did was she went back and deleted these elements one at a time or in pairs. And so she could show that if she deletes one at a time, basically there's no effect on replication timing or one of these small reproducible effects. And actually C element had a pretty big effect on the one side, but she never hit on this randomly with the unbiased deletions that she used. Then she knocked out pairs of them, and you can see even with 2 knocked out, she got still the earliest replicating region was still, this in the whole area was still this domain. So it was still replicating early, so to speak. And she had a 680 kb deletion that took out like all of this stuff all the way up to this yellow and got exactly this replication pattern. So it explained not only the 60 years of inability to find them, that is, they're redundant. Not only was it difficult to make deletions in mammalian cells, but you could make them. It's just if you deleted one element and hit the right element, they're redundant and you wouldn't get any effect. But also it explained all of her complicated results that we were banging our head against a brick wall for 2 years. Okay, so this is my last slide to discuss here. This is how we think things are regulated. It's a working model. We already are finding holes in it, but it's serving its purpose to give us testable predictions. So in this model, here are the ERCs here in colors. They are sites of OCT4-SOX nanog co-binding. So these are the core pluripotency transcriptional regulatory network factors. So what we think is that these represent cell type, and they're really driving the cell type specific replication timing program. And I have other data to support that, but I don't have time to show it to you. But what we believe is that these somehow, there's some sparse data in the literature, but there's nothing really proven, but somehow the OCT4-SOX2 nanog binding allows these regions to interact and recruit histone acetyltransferases, which then make these large swaths of H3K27. And when they interact in 3D space, we now have a hub of histone acetylation. And once we have that, we should be attracting a lot of histone acetylation binding proteins, such as the bromodomain proteins, BRD2 and 4, which have been shown to phase separate. So now we're going to make that microdomain that I talked about in the 90s, or the phase separation that we talk about now in the 2000s. And then Chris Sansam recently showed, he did a two hybrid study to try to get interactors of Treslin. So he used Treslin as the bait, and he pulled out as the top hits BRD2 and 4. So what that means is, and I'm sorry, I may not have said this, but Treslin is a replication initiation protein. Its only known function is to combine with a bunch of other proteins to go in and activate the helicase and initiate DNA replication. So we have a bona fide initiation protein that is likely localized to phase-separated droplets of BRD2-4. And this is speculation, but it's definitely with good premise. And so so this then would form a microdomain that has a very low threshold. As soon as the cyclin-dependent kinases come up at the beginning of S phase, these are going to be the domains that are going to replicate first. And then, as shown here in the cartoon, if you either delete ERCEs or differentiate cells, you disassemble this structure, you move to the periphery, which we've shown, and you assemble heterochromatin, okay, and replicate late. And I didn't say this, but actually when we delete ERCEs, all transcription in this domain goes out. They move from the A to the B compartment. They move to the nuclear lamina, in addition to the early to late replication switch. So what we're trying to do now is bash these up, and we've been able to, in at least one case, uncouple the transcription effect from the replication timing effect, but we're going to find out what we can uncouple and what we can't. Okay, so just to conclude, I'm acknowledging 25 years of people in my lab, okay? And I'm acknowledging the Allen Institute for those live cell images that I showed. And I want to say that I moved last July from Florida State University to SDBRI because I feel like I've done my time with teaching and service to university. I've done a tremendous amount of that, and I want to spend the rest of my career just doing research. And SDBRI is a small, we're currently only 9 faculty, and we want to stay small. It's a small soft money institute. That means we're independent, and we have to pay our own salary, and we have to pay all our own bills, but we can do whatever we want. And we want to stay small so that we don't get a whole lot of bureaucracy going. And I really like this attitude, and that's really how I want to spend the rest of my career. But I want to point out Joanna Davies, a type 1 diabetes person, is the CEO. She started this 7 years ago, and last year we were ranked number 7 in NIH dollars out of 100 funded institutions in San Diego County. So that's pretty good. We're going to stay small, and we're probably not going to bust these guys. But you know, I think that's pretty good. And we don't have a graduate program, so you graduate students out there, please think about coming to San Diego for a postdoc. And that's the end of my talk. Unfortunately, when you're sharing screen, you don't really know what time it is, so I hope I'm on time.
You were perfect. Thank you so much, Dave. Virtual applause to you. And so now we have time, a little bit of time for questions. Again, please type in your questions either directly to me or to share them with everybody. And we can focus on questions for, actually, I think we can open up questions to everybody who presented today. And perhaps I can start. So Dave, I had a quick question for you. Just wanted to clarify. So in your view, these replicating domains are pretty much congruent with TADS. So one of the hypotheses has been, or topic of discussion, whether TADS are kind of dynamic elements that make sense primarily when we look at the statistics of a lot of cells, and whether they actually correspond to physical structures or not. What is your view on this in regards to the stability of the replication domains?
Yeah, so as you all know, when you look in single cells at TADS, they have a lot of heterogeneity. And I would guess that there's a tremendous amount of heterogeneity in replication domains. We know for a fact, quantitatively, in fact, we published just last year in Molecular Cell, how stochastic initiation is. So we know that in different cells, replication is initiating at different sites. And whether those are related to the individual changes in TAD structure in individual cells, that's going to take a lot of work to determine. Let's see, I just sort of download my thoughts on your question. The other thing is that if you align population high C with population replication timing, what you're going to find is that there's a subset of domains that align very, very well, and then a bunch of them that don't align. So so we're still trying to figure that out. I think what I showed you, that one slide I went through extremely fast, what we found was that those particular domains that are dot domains, so if there are dots on both sides of a boundary, you're going to get that really high enrichment of initiation at the boundary. And the boundaries of replication domains in RepliSeq have to have origins at have to be bounded by where the last origin is, because they're all firing synchronously. So as soon as the origins stop firing, the pattern of replication is going to look like forks moving. So that's a good alignment. But there's some alignment of late domains with non-dot domains, but there's not 100% alignment. So there's a lot of things we don't understand. I think those are the two things I want to say. So there's heterogeneity. I think it's going to take a lot of work to look at whether within the same cell, the replication, matches the 3D architecture of that particular TAD. But when you pile up single cell data, you get the TAD back. So there is an average structure. And I'm a big fan of, stochasticity and heterogeneity. So, but in general, Mother Nature gets it done. What's going to be interesting is there might be some outliers, right? Some individual cells where the domain ends up in the wrong place in the nucleus. And that's something we're interested in looking at with those live cell imaging guys, where we could mark the nucleotides that replicated stably and then watch pCNA come back to them and see if there's some guys that just, they don't replicate at the right time in the next cell cycle or the next cell cycle because they're in the wrong place. I don't know if I'm answering your question. Of course, it's a big question.
That's wonderful. And just had another question. So we just listened to a talk by Grey Monroe about evolutionary biology of mutations. So one thing I was wondering about, why do you think the system evolved in a way that we do have early replication, late replication? What are the evolutionary drivers of that?
Yeah, so we had a paper middle of last year where we finally were able to get direct evidence for a model that I've been proposing actually since I was an undergrad. I have a paper from Rick Fertel's class at UC San Diego. I guess that's why I'm back here, because it's my alma mater across the street here. But which is that the reason that there's a temporal order, of course, you shouldn't have to replicate the DNA in a particular temporal order just to duplicate the genome. The reason there's a temporal order is because chromatin is assembled at the replication fork. And therefore, you can drive the assembly of different kinds of chromatin by assembling it at different times and places in the nucleus. Okay, but we never had a way to even test that hypothesis because we couldn't disrupt the timing program. And so in the last several years, we've identified a protein, RIF1. When we knock it out, we get a massive genome-wide disruption of the timing program. And we could show that the timing program is the primary effect, the direct effect of the knockout of RIF1. And then gradually, cell cycle after cell cycle, the chromatin gets incorrectly assembled. And so the timing program is actually necessary for the epigenome. So that's my answer to that question. And I think what it's going to be necessary for is not so much maintaining a cell state. Cells are okay. They live without a timing program, but they die during gestation at different random points. So we think now the hypothesis is that you need that epigenome lock-in mechanism to change cell fate and to really push cells into different, from one cell type to the other. So if we start looking deep into differentiation pathways, lineage commitment, that we should start finding problems, but we haven't got there yet.
So when this timing is disrupted or just becomes completely random, let's say, what happens to the chromatin? How does it look?
Yeah, so it's not completely disrupted. What happens is certain histone post-translational modifications get redistributed. Mm-hmm. So, like for example, H3K27 acetylation, you've got in wild type cells these nice strong peaks that align with the early replicating regions of the genome. And when you knock out RIF1, gradually cell cycle after cell cycle, and it requires entry into S phase. If you hold them in G1, nothing changes. So the primary effect really is replication. But then the replication program is completely disrupted in the first cell cycle. And then these the histone post-translational modifications become gradually redistributed and sort of flattened throughout the genome. Not all of them, and not all peaks. So it's not a complete abrogation of the epigenome, but it's a pretty nasty effect.
Very, very interesting. It's very, very fundamental too. So I have to say that here we assembled, we have really phenomenal speakers, but also leaders in your fields and also thought leaders. So I can't help myself by ask one fairly kind of, I guess, philosophical or speculative question. So in your view, what are the biggest problems that I think you will be in genomics in particular that you think will be able to tackle in the next 10 years, but also the most challenging problems that we'll tackle in the next 10 years? What is the elephant?
Okay.
It's a question to actually everybody. Everybody is welcome to.
Yeah, good. I'm glad you're letting me off the hook on that. Actually, the 4D nucleome just tried to define one single collaborative effort that we all 125 PIs could do, and the Google Doc ended up, with this many questions, and that's the consensus. So there's so many of them. Just what is a loop? We've got to tackle heterogeneity, imaging and omics. Getting imaging to be normalized from lab to lab is a big thing. It's a big effort. And then I think, what is the meaning of the genome architecture? We're still kind of finding anecdotal examples where it looks like it matters. And then lastly, how are we going to apply that to help the human condition? How are we going to help human health, or, if you're interested in the environment or other things, but how are we going to help give back to society with genome architecture? I think these are, that might be the biggest thing, because we're obliged to do that. But somebody else, please.
Yes, let's open
Alistair.
Alistair, Grey, Ursula, please, tell us what you think.
My camera is not connected. There I am. I have a good question. I echo Dave's sentiments on, and maybe not an incredible amount of agreement on. There's some of the parts that resonated with me. I think on the 10-year timeline, I do think we will. A deep, important question is understanding what portions of the genome are, I think, talking to one another in cis, and I think we've actually made remarkable strides on that in the last 10 years, and I do expect to see remarkable strides in the next 10 years. That even today, it is still relatively common to link GWAS hits, SNP mutations that are associated with a disease to try to link to a gene to get the function to choose the nearest gene. And we are increasingly learning better rules than simply linear genomic distance that are improving, and we have an incredibly long way to go. It's in some ways disappointing how much the improvement hasn't made solved all of those problems, but I think we are really tackling that question now, and I do expect we will in 10 years have significantly more robust linkages than simply saying I guess it's the nearest one.
Well, I mean, I think that I don't know how I'm gonna say something that hasn't been said, because I think that there's just some amazing people here today speaking. I guess I'll try to inject a sort of evolutionary biology perspective into this. I mean, I think as an evolutionary biologist, looking at all these talks, I mean, seeing how important it is to understand the sort of epigenome biology as a regulator of processes that have such relevance for evolution. So, I mean, I think one of the big things I anticipate, or I hope for in the next 10 years, is to see a continued synthesis between this kind of fundamental genome biology with evolutionary theory, because I think it's so fundamental that the genome is both the blueprint for the organismal biology, but it's also the unit of heredity. And so there's this biology that can influence heredity by changing things like mutation rate, by in what we just saw from Dave about replication timing, and whether that's also implicated in mutation rate differences. And so are these mechanisms so because mutation is the fundamental source of all genetic variation for evolution, there's just this, I think it's fascinating that it's both, the genome is proving to be both kind of providing the material to dictate organismal fate, but it's also the basis of genetic diversity that can be subjected to evolution. So I think there's this sort of cool circularity that has yet to be kind of integrated into evolutionary theory completely. So I'm really excited to see that continue.
Well, I can't agree with all three of you more. I think these are fascinating questions, and these questions would be on my list too. To our defense, and by us, I mean everybody who is looking at genome structure. How long ago was the structure, the linear sequence of genome understood? I mean, it's depending, when we start counting, it might be, let's say, 50 years ago, 60 years ago, right? And at which point did we get a more or less reliable way of editing a linear sequence? Let's say CRISPR, right? So it did take quite a few decades from the time when there was some basic understanding how the system looks like, to the point when we began to engineer it. And we still can't really create CRISPR babies, right? And then understanding that genome structure plays such a critical role, and we heard today, in so many processes, from the basic evolution, adaptation, mutations, et cetera, and differentiation. That understanding came much later. It came, again, maybe let's say 20 years ago, right? Depending again, when you start counting. So if in the next 10 years, we will learn how to engineer or regulate those systems, we're actually doing quite well compared to what classical genetics did. Would you share this optimism?
I'll share that optimism. I mean, I think that, we've seen a lot of talks, with, I think, getting down to these mechanisms. I mean, being able to, knock out genes and then test stuff, I think that's fundamental, that's a fundamental new tool in the toolkit, so yeah, I'm optimistic.
Well, and on that note, I think before we get all pessimistic, I think that's a good time to conclude the symposium. I enjoyed it tremendously. I would like to thank all our speakers again. This was really a fascinating experience. I love all the talks, and I would love us to stay in touch. And on behalf of everybody at the Physical Genomics Center, on behalf of the Cancer Center and the Northwestern community, thank you so much for being here, albeit virtually. And I hope that next time we can be in person. I have selfish reasons for this, because if we were in person, I would take you out for dinner, and that's when I would start torturing you with many more questions. And we have like a couple of hours talking about questions and asking questions and philosophizing and speculating, and that would be fun thing to do at some point. Thank you so much.
Thank you.
Thank you.
Thanks, everyone.
Thanks for the invitation.
Take care. Thank you.
Yeah, it was great. Good questions. I was going to add optimism actually at the end.
Okay, go ahead.
I was gonna mention that when I was on the job circuit, I interviewed at a company called Inscripta, and a startup company, which I didn't go to because I didn't want to give up my research program and say goodbye to all my students and postdocs. But they were doing fantastic things with gene editing. And I think that the next big wave in gene editing is going to be super